So, hello students, welcome back to your own channel Vedantu NEET English. This is Baswaraj sir, your biology master teacher. So students, let me know in the chat right now if my voice is audible to all of you and the beautiful presentation behind me is visible to all of you. Quickly let me know in the chat if I am audible and visible to all of you. Now students, since NCERT line by line is on the way, right, we have finished major chapters, right? We have finished major chapters like your cell, the unit of life, cell cycle and cell division. And we have also finished the first chapter of plant physiology, that is photosynthesis in higher plants, done and dusted, right? Done and dusted. Now we'll be starting off a new chapter called as respiration plants. And students, all the DNA students watching this lecture, this is going to be a revision for you. So listen very carefully. And all students of YouTube, I will teach you in detail every single line of NCERT. And you should also know, for all the YouTube students also, this is going to be a revision. Why? Because in RNA, we have already finished this chapter in RNA series once. So this is a, what do I say? Line by line is actual revision for everyone. And also you will realize that when we read the lines of NCERT, you will realize that yes, this point is missed out. This point is missed out. So to make sure you do not miss any point, watch the video till the very end. Why? Because till the very end, if you watch, you will be able to solve a lot of questions and you will understand much more details of every single concept. So with that being said, students, quickly let me know in the chat if you are excited for today's class. Quickly let me know. Now, we will start the class by a simple difference, right? Can anyone tell me what is the major difference between breathing and respiration? Everyone in the chat, tell me, what is the major difference between a breathing and respiration? Can anyone tell me? Now you will tell me, sir, breathing is the much more of a physical process. While respiration, right here, respiration is more of a chemical process, right? Breathing is the physical process, respiration is the chemical process. Now I'll ask you one more question. Where does the respiration takes place inside the body of an organism? Where does the respiration takes place? So all of you will tell me, sir, respiration takes place where? Respiration takes place inside the cells. Respiration takes place inside the cells. Okay. Now, since I am breathing here, I am walking, I am talking. Walking, talking, breathing. Does all of this require energy? Now you will tell me, sir, yes, every single function of a human body, it could be movement, locomotion, breathing, excretion, reproduction, every single process requires energy. Right? Requires energy. Now I can ask you one more question. Where is this energy coming from? How am I able to talk so much? Blah, blah, blah from that time. Where is the energy coming from, right? Where is the energy coming from? Now you will tell me, sir, nicely afternoon, I did one nice batting I did. What is the meaning of that? Nicely I did batting means what? Nicely I ate my lunch. Early morning, one full plate of idli. 5-6 idlis, 2 vadas, sambar chutney, nicely wipe the plate out. In the night time, again 3-4 chapatis, 2-3 palya, everything will go inside my body. So can I tell? The source for all this energy is none other than the food which you eat. Now tell me, where is the food coming from? Is someone buying you the food? Obviously, if someone is buying the food, you will eat the food. Free food. Who doesn't say no to free food, right? If you have to buy the food, you'll think twice. Should I spend the money now? You'll think. But let me ask you, where is the entire food in the world? Right, the entire food in the world, where is it actually coming from? Is from the top, is someone throwing the food? No. From somewhere the food has to come, right? The entire food is actually coming from the plants. It's coming from the plants. Okay. Now, how are the plants able to produce the food? Obviously, plants are able to make the food with the help of a process called as photosynthesis. Photosynthesis. Process of called as photosynthesis. Now you tell me, students, in the kitchen and all, yes. You ever got it after DNA class again now? See, Likita, that's why I told you. You don't have to concentrate, just listen to what I'm saying. Listen to what I'm saying. Right, listen to what I'm saying. All of you will easily understand if you just pay a little bit of attention. Okay. Now, Shreyas is throwing from the sky. Shreyas will throw you from the sky now. Wait. Okay. The food. Now you tell me, students, is every single food gives the same amount of energy? You will be like, no. Some food will give you more energy, some food will give you less energy. Right. So every single food provides different energy, different amount of energy, different amount of energy. Different amount of energy is given by every single food. Different form of amount of energy. Now, students, this energy which is given, how will it be used in our body? This energy will be used in our body with the help of a currency called as ATP. ATP. Clear, all of you? Any doubt till here, students? Quickly smash the like button, all of you. All the people watching, don't be lazy. Just take your finger, go click and come. That's all it takes. Okay. Now, students, all the food is given by the photosynthesis used for energy. Now, I'll give you a definition for photosynthesis. So, I'll give you a definition for respiration, or small definition. Understand what exactly is happening in respiration, right? For example, this is a cell right here. For example, this is a cell. Now, in this cell, oxygen is entering. Yes, oxygen entering. In this cell right here, the food is also entering. The food is also entering. Now, tell me, students, this food right here, is it very complex or very simple? Tell me, the food which you eat in your body, is it complex or very simple? You will tell me, sir, this food right here is very simple. So, very complex, sorry. This food right here is very complex substance. It is a complex substance, like carbohydrates, right? Like carbohydrates, proteins, lipids and everything. Complex substance. Okay. Now, when the oxygen food is entering inside a cell, this is a cell. What happens? The food gets oxidized. The food gets oxidized, and when the food gets oxidized, it provides energy. And this energy is stored in the form of ATP. Any doubt till now? This is your entire chapter in a simple format. That is, complex substance is broken into simple substance by oxidizing within a cell to provide energy, and energy stored in the form of ATP. Energy is stored in the form of ATP. So what actually happened here, students? What actually happened here is complex substances broken down into simple substances. Within a cell. Within a cell. With this complex substance, how are they broken down? They are broken down where the CC bond, the carbon-carbon bond is broken. The carbon-carbon bond is broken. That what is the process we call it? The process is called as what? Oxidization. Oxidization is happening within a cell. And when this oxidation is happening, simple substance will provide energy, and that is stored in the form of ATP. Done. This is basically respiration. Nothing very difficult, very easy to understand. Now, students, in the starting of the class, I'll tell you. Tell me, all of you know what is metabolism? Metabolism is divided into two categories. What do we call that? Catabolism, anabolism. All of you agree with me, right? Metabolism is divided into catabolism and anabolism. Now, what is catabolism? Complex substance broken into simple substance is called as catabolism. Okay. What is anabolism? Simple substance is making complex substance, that is anabolism. Now, you tell me, students, currently if you read the definition, can you tell when the complex substances Can I tell when the complex substance is broken into simple substance? So can I call this phenomena? Can I call this process right now as catabolism? Can I call it catabolism? I can call it catabolism right now. Catabolism. Eventually, when the chapter progresses and we reach towards the end of the chapter, that time I will tell you a new definition also. Something called as amphiboic pathway. Amphib. That we'll learn later on. But right now you understand it is catabolism for now. It will change later on. It will change later on. Now students, tell me a process in which energy is released. What is the process called as? Do we call it exothermic or endothermic? All of you in the chat, tell me. Do we call that exothermic or endothermic? This right here, since energy is released, can I call it as exothermic reaction? Can I call this exothermic reaction? Yes. I can call this exothermic reaction. Exothermic reaction. Then similarly, I can also call it a physicochemical reaction. Or I can also call it a physicochemical reaction. Physicochemical reaction. Physicochemical reaction. Why? Because there is exchange of gases. There is a reaction is happening with the help of an enzymes. With the help of an enzyme, it is happening. So it is a physicochemical process. So this is the basics of the entire chapter, respiration. Any doubt till now? Quickly ask me in the chat. Yes, we have complex substance breaking into simple substance within a cell where carbon bond is broken, that is oxidation is happening. Catabolism, exothermic, physicochemical reaction. Physicochemical reaction. Done. If this much is known to you, the chapter we can start now. Basics covering is done. Basics is done now. Okay. Basics is done. Now students, now you need to understand something called as respiratory substrate. For example, I told you, we can take, we can take a food, and from the food item, we're getting the energy. From food, we're getting the energy. Now, what is this food? Right? Food which I'm talking about. The food which I'm talking about is nothing but the respiratory substrate. For example, tell me, in breakfast or lunch, how many of you ate rice? Tell me in the chat, raise your hands quickly. How many of you ate rice and sambar or palav or chitrana, something would have eaten, some rice would have eaten? The rice which you eat, right, the rice which you eat has something called as carbohydrates. Carbohydrates. Yes, the carbohydrates becomes the respiratory substrates. We also have lipids. Lipids, all of you remember students, biomolecules. Lipids are made up of fatty acids, and as well as glycerol. Yes, glycerol. Then we have the proteins, which are made up of amino acids. Apart from that, we also have certain organic acids. They provide the respiratory substrate. All the following different type of substances can provide what? Energy. They can provide energy. Clear? Clear students? Clear all of you? Now, one more thing comes here. What about in the case of minerals? What about vitamins? What about in the case of water? Hormones. All of the following cannot be called as the respiratory substrates because they cannot provide energy. Any doubt till now? Quickly ask me. Any doubt now? So respiratory substrates can provide energy for making ATP later on. Minerals, vitamins, water, hormones cannot give any type of energy. Done. Now students, energy currency of the cell. Now, all your parents get the money in your bank account. All of you know, all the parents get the money in the bank account somehow from the company or wherever they work. Now, instead of company giving the money directly to your parents, they'll give the money to the bank account. And whenever there is a requirement, your parents will use UPI or they'll take the money from the bank directly. Right? For example, right, you are going to buy egg puffs. That is the famous example I always give, egg puffs. Okay. And you try to scan the QR code. You scan the QR code, money is sent to the shopkeeper. But money is also getting cut from somewhere. The money is now getting cut from the bank account. The bank account, the money got cut and they sent it to the shopkeeper. Similarly, inside your body, energy is also stored. Inside your body, energy is stored in the form of ATP. In your body, energy is stored in the form of ATP. Like how in your bank account there is money. In your bank account there is money. In your body there is ATP. Whenever there is requirement, your body is spending the money. What is the money? The ATP. Clear, all of you? So whenever there is a requirement for your body, for example, you have to talk a lot. So more energy is being consumed. And how do you get the more energy? By ATP. So every single time there is a requirement in your body, ATP is being consumed. Every single time you want to go out, eat something nice, drink something nice, juice, what do? You spend the money. The money is the ATP, also called as the energy currency of the cell or the body. Energy currency. Now then what is this NADPH and FADH2? Understand concept, understand. Now all of you know check. Please be careful when you're writing the check, okay? Please be careful. The check, what will happen is, if you take the check and deposit in the bank, the bank people will give you the money. Similarly, in your body, NADPH and FADH2 are just like the check. They are just like the check. Like how if you give the check in the bank, they'll give you the money. In your body, if you take NADH or FADH, you will get ATP. Okay. So how if you take the check and if you give, you will get money. You can take NADH and you can produce ATP from it. ATP. And for example, students, listen very carefully. 1 NADH will give you 3 ATP. 1 FADH2 will give you 2 ATP. Is that point clear to all of you? Stop fighting about egg puff now already. Still the fight about which is superior, egg puff, veg puff, paneer puff, mushroom puff, everything is good. One puff I told you, still fighting about the puff only. I don't know why. Okay. So 1 NADH or FADH will give you what? 1 NADH will give you 3 ATP. 1 FADH2 will give you 2 ATP. So every single time you take the check and deposit in the bank, it gives you money. You take 1 NADH, it gives you ATP. Beautiful. Now students, understand in ATP, where is the energy stored? In ATP, where is the energy stored? Understand that, okay, understand. So here we have ADP, adenosine diphosphate, very important concept, understand. We have adenosine diphosphate. This is your ADP. Di means what? Di means 2. All of you know, di means 2. Di means 2. Diphosphate means 2. Okay. Here we have 2 phosphate. Now, one more phosphate is added to it. We simply cannot take and like go stick there. Can you take this one phosphate, you can stick it, it will not stick very easily. To stick this phosphate to here, we need some kind of glue. We need some kind of Fevicol. We need Fevikwik. Something we need. The Fevicol, the Fevikwik will come in the form of energy. So whatever energy which you get from food, whatever energy you got from egg puff, whatever energy you got from that chicken puff, whatever energy you got from that mushroom puff or paneer puff, puffs, not puff, sorry, very wrong English, puffs. Whatever energy you got from that puffs, right? that energy is used. That energy is used to make a bond here. Bond. That is the Fevikwik here. That is the Fevikwik or Fevicol. And you use that energy. You use that energy and you add the one more phosphate here. So that is how one ADP gets converted to ATP. It's converted to ATP. Clear, all of you? Clear? So ADP is getting converted to ATP by adding the energy here. Now you tell me, all of you. Now you want to remove this. You want to remove this phosphate again. You want to remove this phosphate. How will you remove the phosphate? Tell me. When your hands get stuck in Fevikwik, you keep it in water and you remove it after some time. Now what you do here is, whenever you want the energy back, all you need to do is break the bond here. Break the bond. So I told you, all the energy is stored in the bond. What will happen if you break the bond again? If you break the bond here, obviously the phosphate will come apart. And when you break the bond here, energy will be released. When you break the bond here, energy is being released. And this ATP again gets converted back to ADP, adenosine diphosphate. This is the basics of this chapter. You will understand the importance of this when you do glycolysis. Clear? Yes, breakup is happening. Sorry, one breakup is happening here. Okay. So whenever there is requirement of energy, this bond is broken and the phosphate is released, and with the breaking of bond, energy is being released. Termination from the terminal position, phosphate bond liberated. ATP will become ADP again. Okay. I hope that concept is clear to all of you. Now. About ATP, what is NADH, what is the basics of respiration. Okay. All of you know this much now. All of you are experts now. Full experts you are now. Okay. Now one more question is in front of the board. Let's see who can answer this question now. For example, how are we getting the food? We are eating the puffs, veg puff or paneer puff, something we are eating. But the ingredients for that puff, did it come from the plants only? Yes, it came from the plants. The chicken puff you're eating, the chicken ate the plants and the chicken got energy. Plants is the primary source for everything, right? Plants are the primary source. Plants are the primary source of energy. Now, if I ask you a question, how are the plants getting the energy now? That is, where do plants get the respiratory substrate from? Where do they get it from? Can anyone tell me in the chat? Anyone? Try answering, experts. Try answering. Where do plants get the energy from? Plants doesn't have to do anything. Plants can do a process called as photosynthesis. All of you know that, right? Plants have a simple process called as photosynthesis, in which carbon dioxide is getting reduced. All of you remember, don't forget, photosynthesis and air plants. Plants are able to convert what? Reduce carbon dioxide into glucose. They can reduce carbon dioxide into what? Into glucose. All of you remember there is splitting of water. In non-cyclic photophosphorylation. All of you remember? Splitting of water in the non-cyclic photophosphorylation, in the PS2, water is being split open. All of you remember there is something called as C3 cycle, C2 cycle and C4 cycle. Yes. And all of you also remember that oxygen is the byproduct of, splitting of water. Done. So this is how plants are able to get the food. They're able to take the atmospheric CO2, use the light energy and chlorophyll to convert CO2, reduce CO2 into glucose, and the same glucose is stored in the form of sucrose in the plants. And it is the same sucrose which all of you thieves are going and stealing it from there. Plant is papa. Whole day, whole night it is working. Whole day, whole night the plant is working and it is producing glucose and sucrose. Quietly all of you are going, chop the plant, eat the plant and survive. That's what is happening. That's what is happening. And understood all of you. So the substrate, plants get the substrate from where? They don't get it from anywhere. They make their own substrate. They make their own substrate. Students, remember, it is the same glucose now. It is the same glucose, which is now reacting with oxygen to produce CO2 and H2O as a product. Same glucose, the same food which I spoke here, students. The same food will now enter here. Remember in the starting I told you. Here I told you food is coming from the outside. But in the case of plants, the food is prepared inside. Glucose is produced inside the cell and plants are using this glucose and making their own energy. See, plants are so smart. Plants are independent. Right? Nice. Now students, let's read the lines of NCERT. Ready? Ready all of you? Yes, quickly smash that like button all of you, because this is line to line of NCERT. Every single line will be clear to all of you now. Okay, let's read what is given in NCERT. All of us breathe to live. Obviously, if you don't breathe, you will die. Unless you use oxygen mask, then also you need to breathe. Okay, all of us breathe to live. But why is breathing so essential to life? Can anyone tell me? Obviously, we need oxygen. Okay, what would happen when we breathe? Happens when you breathe. Also, do all living organisms, including plants and microbes, breathe? If so, how? Instead of telling the answer, question they're asking us more and more questions. We'll skip all that. We'll go down. All living organisms need energy for carrying out daily life activities. It could be absorption, transport, movement, reproduction, or even breathing. Where does this energy come from? We know we eat food for energy. How is this energy taken from the food? Oxidation. How is the energy utilized? Formation of ATP. Do all the fluid gain the same amount of energy? No. Do plants eat? Obviously not. They make their own food. Where do plants get their energy from? Photosynthesis. And microbes, for their energy requirements, do they also eat food? Yes, they also eat food. Any doubt in this paragraph? Quickly ask me, students. Any doubt in this paragraph? I think all the answers are, we know these answers now. Next. You may wonder that several questions raised above, they may seem to be very disconnected. But in reality, the process of breathing is very much connected to the process of release of energy from the food. Let us try and understand how this happens now. Let's try. Okay. So before we try and understand that, let's understand the difference between autotrophs and heterotrophs. Very simple difference. The main difference between autotrophs and heterotrophs is that how they obtain the energy in the cell. For example, students, in the case of heterotrophs, oxygen is entering the cell. The food is also entering into the cell. It is getting oxidized. And eventually, after getting oxidized, it is getting converted to energy. And eventually, energy is stored as ATP. Yes. But in the case of plants, students, oxygen is entering, but the food is never entering. In the case of leaf and stem, where photosynthesis happens, tell me, if the photosynthesis is happening inside the leaf only, does the food has to come from outside now? Tell me, if the glucose is produced, C3 cycle is happening in the mesophyll cell. So the food does not have to come from anywhere else extra. So can I tell the food is now produced inside the cell only, Similarly, now what is happening? Oxidation is happening. Oxidization. Yes, which is producing energy. The energy is now converted to, again, ATP. Now you might be wondering, students, what is the meaning of oxidation? What is the meaning of oxidation or oxidization or oxidation? What is oxidation, students? Oxidation in terms of biomolecules or biochemistry is nothing but, let me write in red, full effect will come now. Loss of electrons or loss of protons. So what is actually happening here is the food is getting oxidized. Meaning is that what? The meaning is that from the glucose, for example, glucose is there. The glucose is the bonds are broken in the glucose. When the bonds are broken in the glucose is losing the electrons. It is losing the protons. And the electrons are taken up by oxygen towards the very end. The final electron acceptor is going to be oxygen only. You will understand that in the electron transport chain. All of you are getting an idea about it now? All of you getting a small idea? Yes. So, oxidation is nothing but loss of electrons. Your carbohydrates are losing the protons and electrons. That is taken up by oxygen towards the very end. Okay? Beautiful. Difference is done. Now, students, remember, I wrote here leaf and stem for autotrophs. Why? You tell me. Can the roots do photosynthesis? Can the roots flower? Can the roots and flower do photosynthesis? Roots and flower? Or even the woody stem? Can it do? Tell me. It can't do. Why? Because there is no chlorophyll or chloroplast in the fruit or flower. So in the case of fruit and flower and the woody stem, this mode of nutrition happens. Since the food is not produced, within the cell, in the case of root and flower and stem in some cases, the food has to enter into the cell even in such parts of the plant. That is very important to understand. Concept here. Okay. So when I say autotrophs, it is mostly the green parts of the plant. I hope that that concept is going to your head now, not running over your head. Okay, I hope it is going to your head now. Next thing, let's read what is given in NCERT. Okay, all energy required for life processes is obtained by oxidation. And all of you know what is oxidation? Oxidation of some macromolecules. Students, tell me, all of you know what is macromolecules? Do you find macromolecules in the retentate or filtrate? Do you find macromolecules in the acid soluble or insoluble fraction? Do you find macromolecules in the molecular weight more than 1000 daltons or less than 1000 daltons? All that you should tell me. Quickly go. Tell me in the chat, all of you. What is macromolecules? All the students who have learned biomolecules should be able to answer this very easily. Biomolecules. Where do you find macromolecules? Okay. That we call food. Only green parts, see written here. Only green plant and certain cyanobacteria, because they have chloroplast. Cyanobacteria can prepare their own food by the process of photosynthesis. They trap light energy. Students, tell me, all of you who learned photosynthesis, this trapping of light energy, how is it possible? Tell me, all of you. This trapping of light energy, is it possible because of certain pigments like your antenna pigment and reaction center? Yes or no? Quickly tell me in the chat. Yes or no? Yes. And convert it to chemical energy. Students, tell me, this conversion of light to chemical energy, does it happen in light reaction or dark reaction? Answer in the chat. Light reaction or dark reaction? That is stored in the bonds of carbohydrates. See, it is stored in the bonds of carbohydrate like glucose, sucrose and starch. We must remember that green plants too, not all cells, very important line, not all cells, tissues and organs photosynthesize. Only cells containing chloroplast that are most often located in the superficial layers, leaf and stem, carry out the photosynthesis. Hence, even in green plants, green plants, all the other organs, tissues and cells, root, flower, woody stem that are non-green need food for oxidation. They need food from the outside to inside. Outside to inside. Here. We learned it here. Okay. So not all green, only the green parts of the plant can do photosynthesis. Not everyone. It is not everyone's cup of tea. Okay. Hence, food has to be translocated to all non-green parts. All of you learned that. Animals are heterotrophic. All of you know. That is, they obtain the food from plants. Directly herbivorous or indirectly carnivorous. Tell me in the chat, how many of you are herbivorous and how many of you are omnivorous in the chat? All of you tell me in the chat also quickly. How many of you are herbivores? How many of you are omnivores? Okay. I believe no one is carnivorous and all. Okay. No one is carnivorous. Now, saprophytes, all of you know the fungi, dead and decaying matter. What is more important to recognize is that ultimately all the food that is respired for life process, all the food respired for life process comes from the photosynthesis. Now tell me, students, will there be life on the earth? Will there be life on the earth if photosynthesis stops happening? Tell me. Will there be life on the earth if all the photosynthesis stops at one go? Obviously not. Okay. So this chapter deals with the cellular respiration, the respiration happening within the cell, or the mechanism of breakdown of food material within the cell to release the energy and trapping of this energy to synthesize ATP. Photosynthesis, of course, takes place within the chloroplast. All of you know that. Whereas breakdown of complex molecules to yield energy takes place in the cytoplasm. So if I ask you a question, where is respiration happening? Respiration is happening in the cytoplasm and mitochondria. All of you type in the chat. All of you type in the chat. It is happening in where? Respiration is happening in cytoplasm and mitochondria. Photosynthesis is happening in the mesophyll cells, that is chloroplast. Okay. See, respiration is happening where? Respiration is happening in cytoplasm and mitochondria. Okay. Definition for all the board students. Definition here. Definition time. The breakdown of CC bond of complex components like glucose, right? Complex compounds through oxidation, loss of electrons, through oxidation within a cell, see, very important, within a cell, leading to release of energy, leading to release of considerable amount of energy is called as respiration. It is called as respiration. So breaking of CC bond of what? Breaking the CC bond of glucose through oxidation. How? Through oxidation within a cell, leading to the release of considerable amount of energy is called as respiration. The compounds are oxidized during the process are known as respiratory substrates. Respiratory substrates are the substance from which you derive the energy. Okay. Now, carbohydrates. Respiratory substrate, your anna sambar. Okay. Carbohydrates are oxidized to release energy, but proteins and fats, and even organic acid can be used as respiratory substrate in some plants under certain conditions. Okay. Now, during oxidation within a cell, all the energy contained in respiration substrate is not released in free into the cell or in a single step. Understand this concept. Very simple concept is this. Understand this concept. That is, all of you know, all of you are aware, right? All of you are aware. If I give you, wrong example again. So, students, remember, if I give you to study physics, chemistry, and biology, all the 3 NEET syllabus in one day, will you finish it? Tell me, students, will you finish the entire syllabus Will you finish the entire syllabus in just one day? No, you will not. You will take entire 1 year time. Step by step you'll finish your syllabus. Step by step, you will go further. Every single month you will cover backlogs. You will become more confident and then you will give your examination. In one day you cannot finish. Similarly in the case of cell. Similarly in the case of cell also. The entire energy cannot be contained at one go. It cannot take the entire energy from the food and make ATP out of it. No, it's not possible. Not possible at all. Not possible at all. So what will cell do? Cell will break down the energy. It will use the energy in a stepwise manner. Stepwise manner. That is why students, they say glycolysis is a 10-step process. Glycolysis is a 10-step process because in every step by step, energy is being converted into ATP. So just like how you cannot study the entire NEET syllabus in just one day, all the energy cannot be converted into ATP in one go. It happens in a step-by-step manner. Understood all of you? See here. Respiratory substrate is not released in free in a cell or in a single step. It is released in a series of slow steps. See, series of slow stepwise reactions controlled by the enzymes, and it is trapped as a chemical energy in the form of ATP. So all the energy is not released at one go. One by one, it is produced and released. Okay. Hence, it is important to understand that energy released by oxidation in respiration is not, rather cannot be used directly. It cannot be used directly. It is too much. Energy is too much. It cannot be used directly. So all the energy is stored. All the energy is stored in the form of ATP. All the energy is used to make ATP first of all. All the energy is used to make the ATP. Clear all of you? Tell me. Which is broken down whenever a requirement is there. Whenever requirement is there, ATP is broken down whenever or wherever energy is needed to be utilized. Hence, ATP acts as the energy currency of the cell. ATP acts as the energy currency of the cell. This energy in ATP is utilized in various energy requiring process of organism. And carbon skeleton, see, this is very important line of NCERT. Very important line of NCERT, understand all of you. Focus. Carbon skeleton produced. Carbon skeleton produced. What is carbon skeleton? The carbon skeleton they're talking about here is, tell me students, what is the products of your respiration? We know generally what happens is we have glucose plus oxygen will give you CO2 plus water. This is a general reaction. This is a general reaction. The carbon skeleton they're talking here. The carbon skeleton they're talking here is nothing but the CO2. They're talking about is CO2. Understand? The carbon skeleton produced during respiration is used as a precursor for biosynthesis of other molecules in the cell. What is the meaning of that line? The meaning of line is that this carbon skeleton right here, is used by the cell itself for one more process called as photosynthesis. All of you know that this CO2 can actually take part in photosynthesis. This CO2 again can be converted back to glucose. And this glucose can again be used here. So can I tell students, plants are independent. They can take care of themselves on their own. They don't actually require human intervention or any other organism intervention. Most of the plants can survive on their own. They're independent on their own. Yes. So be like plants. That is my new motto for all of you. Be like plants. Okay. Be like plants. So beautiful. See, this is the beautiful story of plants. Plants will use glucose to use oxygen to produce the CO2. This oxygen is also coming from the photosynthesis only. Yes. This water is coming from the outside. All of you know that. Now this CO2 can again be used in photosynthesis. So plants are very independent. Be like plants. Okay. Now students, we will learn something about aerobic respiration and anaerobic respiration. All of you should know this by now. What is the meaning of aerobic? Sony, why is there a phone call emoji also? I don't understand. Whom you're trying to call, huh? Sony has told be like plants. Sun, blue heart, a call emoji. Then sun, blue heart again. From nowhere a call emoji is coming. Nice. Okay. Tell me, what is aerobic respiration? Aerobic respiration takes place in the presence of O2. In the presence of oxygen. Anaerobic respiration, absence. In the absence of O2. Absence of O2. Okay. And where does aerobic respiration takes place? Aerobic respiration takes place in the cytoplasm. Cytoplasm as well as mitochondria. All of you learned that. Mitochondria. What about anaerobic respiration? Anaerobic respiration happens only in the cytoplasm. Only in the cytoplasm. Only in the cytoplasm. Now what about energy? Here in aerobic respiration, lot of energy is produced. Lot of energy is produced. In the case of anaerobic respiration, less energy. Like how all of you have right now. No energy only in the chat. What is this? Huh? No energy in the chat only. What is this? No energy in the chat. Show some energy in the chat, students. Make everyone know that yes, today you have a class from Baswa sir and you are learning something new. Show the energy in the chat. Don't be anaerobic respiration. Do aerobic respiration, all of you. Obviously, all of you are doing aerobic only, but I'm telling you. Okay. Lot of energy is released. What are the products here? The products, the byproduct. Byproduct is going to be CO2 plus water. Byproduct is going to be CO2 plus water. Okay, CO2 plus water. In the case of anaerobic respiration, you will see something called as ethanol. Ethanol or lactic acid. Or lactic acid. Lactic acid. Okay? Yes. Now random emojis are coming. After I told emoji, some random emojis are coming. Sir, Benton capture energy. Like full random emojis now. Oh God, random emojis are coming now. Someone is putting a sun, someone is putting a clock. I don't know what is clock. Why is clock there? Benton, no. Sir, how long the session goes on? Tanya, it's going to be a long session because we are doing line-by-line of NCERT. It will take some time. Smash my phone now. Someone is too rich. Please donate your phone to someone. Okay. So in anaerobic respiration, students, under anaerobic respiration, you have something called as alcoholic fermentation and lactic acid fermentation. Now you will be learning both of them in today's class only. Today's class will be learning something called as alcoholic fermentation and lactic acid fermentation. Okay, clear all of you? Both will be happening in today's class only, so don't run away anywhere later on. It is very important. Okay. Now, let's read now. Let's read. Do plants breathe coming here only? 1 second, students. Okay, let's read this. Now, students, tell me, do plants breathe? Tell me, all of you. Offline is a peace. Everything is peace in life. No difficult. Tell me, students, do plants breathe? All of you. Ask your plant friends. Go ask your plant friend. Do they show breathing? Breathing and respiration. Do they show? So let me tell you, students, this is a very controversial thing, right? It's a very controversial thing. Plants actually don't technically breathe. They don't, you can't see them breathing. You cannot see in the night time a plant is going hiding behind your back. It is opening its nose and breathing. It will not happen. Behind your back, they're not breathing. Please be assured. But they need oxygen. They need oxygen for their survival. They need oxygen for their survival. If there is no oxygen, they cannot survive at all. Okay. So here, Well, the answer to this question is not quite so direct. See, not so quite so direct, because even NCERT does not know. Okay, okay, let's see now. Yes, plants require oxygen for respiration to occur, and they also give out CO2. All of you know they give out CO2. Yes, hence plants have a system in place that ensures the availability of oxygen. It'll make sure that oxygen is there. Okay, plants, unlike animals, have no special organs. Very important line. This is a PYQ, all of you students. This is a PYQ right here. Unlike humans, like how we have a nose, right? How we have nose, plants don't have a nose to breathe in. Like how fish has gills, plants don't have gills to breathe in, right? And there are certain frog which can breathe through their skin. Plants can't do that also. So if the plant does not have any major organ, how do you think the plants are breathing? Can anyone tell me? How are plants able to take in the oxygen? Yes, all of it, tell me in the chat. So there are two main ways how the oxygen enters basically. The first one is called as stomata, and the second one is called as lenticels. First one is called as stomata, second one is called as lenticels. With the help of these two organs, they can actually exchange of gases can happen. Exchange of gases can happen because of stomata, because of stomata and lenticels. But students, understand, the stomata and lenticels are just a minor organ. They are not specifically made for breathing. They are not specifically made for taking in oxygen. The main purpose of stomata and lenticels is exchange of gases, and also in the case of stomata is transpiration. Yes, all of you. Transpiration, loss, removal of water vapor. But unlike humans, how we have nose, we have lungs, we have trachea, right? We have specific organs. Plants do not have a specific organ like that. Plants do not have a very specific organ like that for gaseous exchange. No, it's not there. Not there at all. Not there at all. Right? But they have stomata and lenticels for purpose. There are several fact reasons why plants can get along without a respiratory organ. That is, plants can survive. Plants can survive without a respiratory organ. They're like, no, not required for me. I don't need respiratory organ. I can take care of my own needs. So students, tell me in the chat right now. Tell me in the chat right now, every one of you. Why do you think plants don't require a special organ? Anyone in the chat? Quickly go. Time starts now. Time starts now. Quickly go in the chat. Why do you think plants don't require a very special organ system for respiration or for breathing or taking in oxygen? Let's see how many of you can tell. Everyone. Quickly go. Time starts now. Yes. Yes, Tanuja. Yes, Sony. Yes, Rashmita. Yes, Muskan, Madhumiti, Sony, Harish Shrine. All of you tell me. And all the DNA students also tell me. Why plants do not require a special organ? The answer is very simple. First of all, very simple. Every part of the plant can take care of itself. It can take care of O2 demand. Can take care of O2 demand. Now, how can they take care of O2 demand? Why? Because students, remember, in the case of plants, O2 demand is actually very less. O2 demand is very less. Even if there is a demand, students, even if there is a demand, all of you remember that even if there is a demand, right? Even if there is a demand, all of you remember, in the case of plants, there is a process called as photosynthesis. Yes, photosynthesis. In this process of photosynthesis, can I tell O2 is a byproduct here? Byproduct. So can I tell students, since demand is very less, Each part is taking care of itself. This oxygen itself can be used by the plants. Tell me yes or no. Tell me yes or no, students. This particular O2 can take care of itself. Done. Second point. The third point is very simple again. Third point is very simple again. I'll show you with the help of a diagram the third point. Understand. This is the stem. Woody stem. This is the woody stem. And in the middle, we have the thick dead cells in the middle. All the cells which provide mechanical support are on the inside. In the case of stem, students, in the case of stem or the branches, all the living cells are present on the periphery. All the living cells are on the periphery here. Can you see? All the living cells are on the periphery like this. Now you tell me, students, on this periphery, we have something called as lenticels. Lenticels here. Lenticels are here. With the help of lenticels, oxygen is entering inside. Since oxygen is a gas, can it diffuse very easily? Yes. Since oxygen is a gas, it can diffuse very freely here. So, since all the living cells are on the surface, oxygen can freely diffuse on the surface. It can freely diffuse on the surface. This is the third point right here. Third point right here. Understand lines of NCERT now. First, each plant part can take care. Each plant part can take care of the own exchange needs, own gaseous needs. Each part can take care of its own gaseous exchange needs. Okay. There is very little transport of gases from the one part to the another. Second, plants do not present great demand for gas exchange. So demand only is very less. Demand itself is very less. Oh, what happened? Demand also is very less. Okay. Root, stem and leaves respire at the rate far lower than the animals do. So demand is very less here. Only during photosynthesis, a large volume of gas exchange and leaf is well adapted to take care of its own needs. See, only during photosynthesis, there is a large volume is required. Only during photosynthesis, large volume is required. And all of you know, if photosynthesis is happening, oxygen is a byproduct. Yes, see here, of gases exchange needs, and leaf is well adapted to take care of its own needs during these periods. Yes, it can take care of its own. Oxygen is there. When photosynthesis, availability of O2 is not a problem in these cells, since O2 is released within the cell. O2 is released within the cell. Second point here. Second point is right here. Now third point. Third, the distance that gases must diffuse evenly, even in large bulky plant, is not great. They don't have to travel a lot of distance. Why? Each living cell in plant is located quite close to the surface of the plant. Told you, each living cell is close to the plant, quite close to the surface of the plant. This is true for leaves. You may ask, what about the thick woody stem or root? In stem, the living cells are organized in the thin layer inside and beneath the bark. They also have openings called as lenticels. These cells in the interior are dead and provide only mechanical support. Thus, most of the cells plant have at least a part of their surface, in contact with the air. Cells contact with the air. Can you see? The air. This also is facilitated by loose packaging of parenchyma within the leaves and stem and roots, which provide an interconnecting network of the airspace. Clear, students? Any doubt till now? Students, you're understanding every single line of NCERT. Every single line of NCERT is being covered. Nothing has been missed. Okay? Any doubt till now? Quickly ask me in the chat, all of you. Some OBI is saying thank you, Bhaiya. What? Or my eyesight is very poor. I can't read properly. Any doubt here? Quickly ask me in the chat. Beautiful. So, three main reasons why plants do not require a special organ. Three main things. Now, students, now All of us been reading this, right? All of you have been reading this particular statement, right? If the energy is energy to be useful to the cell, it should be available to utilize to synthesize other molecules that the cell requires. The strategy that the plants use is in the catabolism of the glucose molecule in such a way that not all liberated energy goes out as heat. See, they're making sure not all energy is used as heat. The key is to oxidize the glucose in not one step. I told you, it does not happen in one step. The oxidation of glucose happens in multiple steps. Like how you cannot read the entire NEET syllabus in just one day, you also do it in stepwise manner. Glucose is being oxidized in a stepwise manner. Can you see? Several small steps enabling some steps to be just large enough that the energy released can be coupled to ATP synthesis. How this is done? Essentially the story of respiration. So that's what you'll be learning, students. That's what you'll be learning. In respiration, you will see, all of you will see when I teach you glycolysis today, it is a 10-step process, 10 different steps. And I will teach you in a manner which you can remember and understand and not by heart it. Okay? All the DNA students already know it because I taught you recently, right? So we will learn the similarly now, students. We'll learn glycolysis also. So any doubt in this paragraph also, let me know in the chat, all of you. Shoot. So all of you should know respiration happens in a stepwise manner, not in an entire large big step. Okay. During the process of respiration, oxygen is utilized and carbon dioxide, water energy is released. All of you know this, right? Energy is being released. All of you are aware of this. We learned here only. See, carbon dioxide, water and energy is being released outside as a byproduct. Okay. The combustion reaction requires oxygen, obviously. Okay. But some cells live where oxygen may or may not be available. Here they are talking about what? Here they are talking about anaerobic respiration. Okay. There are sufficient reasons to believe that the first cell on the planet lived in the atmosphere that lacked oxygen. So basically telling you is the first formed cell was in an atmosphere which does not have any type of oxygen. So what we are trying to understand here is, students, the process of the first step, students, the first step in glycolysis, the first step in respiration, which is glycolysis, it does not require oxygen. The first step in respiration, students, very important line, understand. The first step in respiration does not require oxygen. First step in respiration does not require oxygen. It does not require oxygen at all. We can see your messages. Don't worry. Okay. The first step in respiration does not require oxygen. So can I confidently tell the first step in respiration, which is glycolysis, can take place in aerobic respiration as well as anaerobic respiration. In both the process, in two different types of respiration, glycolysis can take place. Glycolysis, which is the first step in respiration, can take place, does not require oxygen. So it can happen in both aerobic respiration as well as anaerobic respiration. In both the cases, it can happen. Clear, all of you? Clear? Understanding that this is the basic concept which you need to understand before you start glycolysis. Okay? Basic concept you need to know. All of you should know. Okay? See here. Even among the present day living organisms, we know of several that are adapted to anaerobic conditions. Yes, we know many anaerobic conditions, bacteria, we know, right? Some of these organisms are facultative, means flexible. Facultative anaerobes means they can survive in anaerobic conditions, but they can also survive in aerobic also. Okay. While others are anaerobic conditions is obligate. What is the meaning of obligate? The meaning of obligate is strict. Obligate anaerobes can survive only in oxygen condition. Only can survive in an oxygen condition. Obligate anaerobes, they don't require oxygen at all. If oxygen is entering, they will die. Like they're opposite of all of us. And for your obligate anaerobes are opposite of us. Like how we cannot survive without oxygen. If oxygen is entering there, they'll die off. They can't take oxygen. Like, no, stay away from me. 10 feet, stay away from me, oxygen. They're saying, okay. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose. So from here, this line we understand. From this line we understand glycolysis is partial oxidation of glucose, not complete oxidation. Clear? Again, very important line of NCERT. You might skip this line of NCERT if you don't read it one more time. Glycolysis is a partial oxidized glucose. So in glycolysis, there is partial oxidation of glucose, not complete oxidation of glucose. Tell me, students, if you understand that line quickly, this line right here, green as in the chat. Green as in the chat if you understand that line. So in the case of glycolysis, there is partial oxidation of glucose, and glycolysis can take place in both aerobic and anaerobic organisms without the help of oxygen. This breakdown of glucose to pyruvic acid is called as glycolysis. It is called as glycolysis. Clear, all of you? Clear? Clear? Clear, all of you? Yes. That is called as partial oxidation of glucose. You might ask me, sir, what is the meaning of partial oxidation? Friends, in the partial oxidation of glucose, all of you know, in oxidation, your glucose is getting converted to CO2 plus water. Right? All of you know, your glucose is getting converted to CO2 plus water in the presence of oxygen. When this happens, it is called as complete oxidation. Complete oxidation. It has to, your glucose needs to get converted to CO2. Complete oxidation. This is complete oxidation. But in the case of partial oxidation, there is no CO2. There is no CO2 evolved. You will notice this. When you learn the glycolysis, there will be no CO2 coming out. Any step of glycolysis, if you take any step of glycolysis, there will never be CO2 evolution. Is that point clear to all of you? Tell me. Is that point clear to all of you? There will be no CO2 evolution. That is the meaning of partial oxidation of glucose, and that is happening in glycolysis. Students, glycolysis. Any doubt till now, quickly shoot in the chat. I will give you exactly 1 minute to ask your doubts, and then I'm going ahead. Beautiful lines of NCERT, students. All the students who are reading the NCERT for the first time, you will be understanding all these concepts. See, you might skip all this. So I have highlighted important points for all of you here and there. So focus on these points. You will get the depth and clarity on all these concepts today. Okay? Yes. How will I attack you? Tell me. With so much of intense and love for this topic I'm teaching all of you. Hopefully all of you can show the same amount of appreciation for all these concepts, right? Appreciation for all these concepts. because it is very easy to by heart all this. Let me tell you confidently, it is very, very easy to by heart all this. For example, if you go on the internet anywhere right now, if you type trick to remember glycolysis in 5 minutes, if you type on the internet trick to remember glycolysis in 5 minutes, you will get a trick. I am very sure you will get a trick to remember the glycolysis, and you can by heart all entire steps in a mere 15 minutes, and you'll be like, I know glycolysis. You'll nicely joy and run away. Okay, nicely happy and you'll run away. But students, let me tell you, this is what used to happen with me also. When I was your age, honestly, I used to mug it up. There was a very high chance that even all of you would have mugged it up, right? Very high chance that all of you mugged up the entire glycolysis 5 minutes trick, like 5 minutes hack. Okay, 5 minutes trick to learn the glycolysis. And all of you would have mugged it up. But let me tell you, that is not the way we do in my class. That is not the way we do in my class. I will make you understand the depths of glycolysis. I'll make you understand every single step of glycolysis. I'll make you understand and appreciate from your end. You will appreciate every single step of glycolysis by understanding it, but not by hearting it. Okay? Ready? Exactly. You will learn it in 5 minutes, you will forget in 5 minutes. Okay? You will understand in 5 minutes, you will forget in 5 minutes. So, with that being said, can we start glycolysis now, students? I want ultimate focus for the next 45 minutes. Next 45 minutes, I want ultimate focus from all of you. Ignore everything around you, all of you. Ignore everything around you. Just focus and learn what I'm teaching you right now. Make small short notes here and there if you want. But take away this strong topic from my class today. And for the rest of your life, you will thank me. Even when you become a doctor, when you will study biochemistry, and when you learn glycolysis again in MBBS first year, that time also you will appreciate the glycolysis taught by Baswa sir. Okay? You will understand the importance of it. Ready all of you? Tell me in the chat. Ready all of you? Time to study glycolysis. Time to study glycolysis, all of you. Tell me, ready? Now, what is the meaning of, first of all, let's define glycolysis. Let's define glycolysis, all of you. Define the glycolysis, all of you. Okay. Glyco means sugar. Glyco means sugar. Sugar or glucose. Sugar or glucose. Lysis means breakdown. Breakdown. So what is glycolysis? Glycolysis is nothing but the breakdown of sugar. Breakdown of sugar is called as glycolysis. First of all, now we will understand the location. Location of glycolysis. The location of glycolysis is the cytoplasm. Cytoplasm is the location. How many steps? There are in total of 10 steps here. In total of 10 steps. This is called as partial oxidation. Partial oxidation of glucose. Why? Because there is no CO2. There is no CO2 evolved. No CO2 evolved. Partial oxidation of glucose. Partial oxidation of glucose. Where do you find it? We find this particular glycolysis in prokaryotes, eukaryotes, animals, plants, aerobes, anaerobes. In every single organism, we find this process called as glycolysis. Every single organism. Now students, since I'm teaching you botany, let's focus on plants. What exactly happens in the case of plants? Focus. In the case of plants, all of us know as a result of photosynthesis. As a result of photosynthesis, in the case of plants, glucose is produced. Glucose is produced. This glucose will be stored in the form of sucrose. Sucrose. In the case of plants, whenever the glycolysis has to take place, this sucrose is again converted back to glucose. This sucrose right here has to be converted back to glucose when? During glycolysis again. Glycolysis. And the enzyme here involved is called as invertase. Invertase enzyme. Invertase enzyme plays a major role in conversion of sucrose into glucose. And all of you know this sucrose right here is a disaccharide. Sucrose is a disaccharide made up of glucose plus fructose. So what do we learn here? Photosynthesis, glucose is made, glucose is converted to sucrose, that is stored as glucose, stored as sucrose. Sucrose is again converted back to glucose in glycolysis. Enzyme is invertase. Done. First step is done. Now students, I will teach you again the small concept which I have told you repeatedly over and over throughout the NEET journey. And in this step, all of you should know what is first of all meaning of reduction. What is the meaning of reduction? The meaning of reduction is that gain of electrons or gain of protons. Now, what is the meaning of oxidation? The meaning of oxidation is that loss of electrons or loss of proton. This should be very clear to all of you. Now. What is the meaning of reduction? What is the meaning of oxidation? The simple thing should be known to every single student before you understand or try to even understand glycolysis. If you just by heart, don't need to learn all this, honestly. Okay. Now we will understand the other name of glycolysis in which question can be asked from. The term glycolysis can also be called as, EMP pathway. Now you might ask me why? Because students, glycolysis was discovered by three different scientists. Called as Emden, Marinoff and Parnas. Here the first letter of the second name, E from EMP pathway, E from Emden, M from Marinoff and P from Parnas was taken and we call it EMP pathway. We call this EMP pathway. Three different scientists who discovered the glycolysis. Clear? All of you, I think all of you are following till now. Okay. Now students, the entire glycolysis can be divided into two stages. Like how right now you're in a NEET journey, you're preparing for NEET examination and then you will give NEET examination on one day. Right? The cell also, before it undergoes the entire producing the energy, it has to go preparatory stage. So in the case of glycolysis can be divided into two stages. One is called as preparatory stage, other one is called as payoff stage. Preparatory stage, payoff stage. Two different stages in the glycolysis. Okay. Now we will focus on preparatory stage. Now we'll focus on the preparatory stage, step 1. Step 1 of preparatory stage. Now, all of you know, I told you already, glucose is taking part in the reaction. Glucose is entering. Now students, the first step in glycolysis is called as phosphorylation. It's called as what? Phosphorylation. The first step, phosphorylation. Now what is happening in phosphorylation? Understand. Here we have an ATP here. Here we have an ATP. Now students, I told you, in ATP, all of you tell me students, in ATP, where is the energy stored as? Tell me. In ATP, energy is stored in the bonds. Tell me students, all of you remember, energy is stored in the bond, and what will happen if I break this bond right here? What will happen if I break this bond right here? If I break this bond right here, can I tell energy will be released? Can I tell energy will be released from here? Energy will be released. And at the same time, the phosphate will also be released. Two things are released. Yes, it is released. Now what is happening here is, using the energy released, this phosphate right here is attached to the sixth position of the glucose. Sixth position of the glucose. And all of you know, all of you are aware, glucose is 6 carbon, right? That is C6H12O6. So this phosphate right here is attached to the sixth position of glucose. Now you tell me, if I add glucose to the sixth position, can I tell glucose is now becoming glucose 6 phosphate? Why phosphate? Because at the sixth position we have a phosphate now. So in the first step, which is called as phosphorylation, ATP is being consumed, bond is broken, energy is released. Using the energy, one ATP is attached to the sixth position of the glucose, and hence we are calling it glucose 6-phosphate. And in the first step, the enzyme involved here is called as hexokinase. See students, you don't have to remember all the enzymes, at least learn the first enzyme and the last enzyme if possible. Don't forget all the middle enzymes. So glucose is being converted to glucose 6-phosphate in the first step. Now tell me students, if I am removing the phosphate from here, can I tell ATP is getting converted to ADP? Adenosine triphosphate is getting converted to adenosine diphosphate. Done. So that is the first step in glycolysis, which you don't need to by heart anymore. So glucose to glucose 6-phosphate, first step. Glucose to glucose 6-phosphate using a ATP, phosphorylation is happening. Glucose to glucose 6-phosphate. Understood all of you? Understood all of you? That is why it's called as glucose 6-phosphate. Now understand. I will explain every single step. Every single step I'll explain now. Understand. Glucose 6-phosphate. Now, understood, glucose 6-phosphate is done. Now, all of you know what is isomers. Isomer. In chemistry, you might have learned isomer. The meaning of isomer is that they have the same chemical formula. The structure is different. They have the same chemical formula, such as your glucose 6-phosphate and fructose 6-phosphate are isomers. They have the same chemical formula, C6H11O9P. Same chemical structure, same chemical formula, but structure is different. If you notice at the glucose, it has the aldehyde here, and if you look at the fructose, it has a ketone group here. Aldehyde group, ketone group. Isomerization. They can change their shape, change the shape. Same but different. Exactly. Same but different. So can I tell, in the next step, glucose 6-phosphate is getting converted, isomerized to fructose 6-phosphate. And always remember, whenever there is isomerization, there is no release of energy or absorption of energy. No intervention of energy at all. So, what do we have in the second step, students? In the second step of your glycolysis, we have the isomerization, where glucose 6-phosphate is converted to fructose 6-phosphate. Fructose 6-phosphate. Understood? Different structure. Second step is done. Any doubt till now, quickly ask me. I know all of you are on the same page with me now. All of you are on the same page now. In the third step again, students, there is something called as isomerization is happening again. Not isomerization, sorry. In the third step again, phosphorylation is happening again. Third step, phosphorylation is happening again. So again I am bringing an ATP now. Again I am bringing an ATP. Now tell me students, again if I break the bond here, can I tell energy can be released? Third step, energy. Can I tell this phosphate is being released? Yes, phosphate is being released. But this time, this phosphate and this energy is not added to the sixth position. But this energy and phosphate is used to attach it to the, making a bond to the first carbon. See here. This phosphate, by using the energy, it is added to the first carbon. Phosphate. Now you tell me, chemistry, all of you tell me the chemistry, basic chemistry, you tell me. If fructose 6-phosphate, if I am adding one more phosphate to the first position, what will it become? Fructose 6-phosphate, can I tell, is now getting converted to fructose 1,6 bis. Why bis? Because 2-phosphate and phosphate. So your fructose 6-phosphate is getting converted to, is phosphorylated to fructose 1,6 bisphosphate. 1,6 bisphosphate. Did all of you understand the technicality here? You might have just by-hearted till now, that, okay, fructose 6-phosphate, fructose 1,6 bisphosphate. And you're by-hearted why ATP is used here. But now you know why, how you are obtaining the fructose 6-phosphate while using the ATP. Tell me, now if ATP is being used here, can I tell this ATP now is getting converted to ADP? Yes, ADP. Do you see that happening right here in the table? Yes, we see that happening in the table. So glucose 6-phosphate, fructose 6-phosphate is getting converted to fructose 1,6 bisphosphate. 1,6 bisphosphate. So right now we have this structure right here. Fructose 1,6. First position, sixth position. 1 phosphate has been attached. Fructose 1,6 bisphosphate. Done. All of you understood till now? Yes. So what have we covered now, students? Till now we have understood glucose is getting converted to glucose 6-phosphate, phosphorylation, ATP is being used here. Then isomerization, glucose to fructose, same but different. Then we also learnt again in the third step, fructose is again phosphorylated, ATP is being added, and we get fructose 1,6 bisphosphate. 1,6 bisphosphate. Now students understand, critical step is coming. Critical step. Your understanding should become more clear about glycolysis. Understanding. See this. Now tell me, how many carbon is fructose 6-phosphate? Your fructose 6-phosphate right now is 1, 2, 3, 4, 5, 6. 6 carbon right here. 1 phosphate is here and 1 phosphate is here. 6 carbon molecule. Yes, 6 carbon molecule. Now tell me students, what will happen if I break this 6 carbon molecule into 2 molecules of 3 carbon? If I break it into half, do I get something like this? Do I end up getting 3 carbon here? 1 molecule like this, 3 carbon. Other molecule is also 3 carbon. Here we have 1 phosphate. Here I have 1 phosphate. Do I get something like this now, students? Tell me, yes or no? If I break it now. The 6 carbon molecule can be broken into 3 carbon molecule. That is what we are doing in the next step. We are breaking it into half. So if you notice here, till now here we did. Now this fructose 1,6 bisphosphate is breaking into 2. 1 is coming here. A 3 carbon molecule. 2 of them. See here. A fructose 1,6 bisphosphate is breaking into something called as PgAl. Can also be called as gap. What is this gap? Gap is nothing but glyceraldehyde, G, aldehyde, A and P. Gap. Glyceraldehyde 3-phosphate. It can also be called as PGAL, phosphoglyceraldehyde. 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 So meaning is same. Meaning is same. This can also be called as triose phosphate. Triose phosphate. Because your NCERT is mentioning triose phosphate. Can you see? Triose phosphate, glyceraldehyde 3-phosphate in your writing section. When you the text is there, in the text it is writing phosphoglyceric acid, phosphoglyceric acid or phosphoglyceraldehyde, phosphoglyceraldehyde in the written part. So don't get confused. Meaning is same. Meaning is same. Okay. So we are getting first one. The first molecule right here is called as what? The first molecule right here is glyceraldehyde 3-phosphate called as GAP. It can also be called as PGAL, phosphoglyceraldehyde. It can also be called as triose phosphate. Triose phosphate. The first 3 carbon here. First 3 carbon. This one. This one. Now what about the other one? The other one is called as DHAP. DAP. What is DHAP, students? Understand. DHAP is nothing but dihydroxy dihydroxy phosphate. Dihydroxy acetone phosphate. Dihydroxy acetone phosphate. DHAP. Dihydroxy acetone phosphate. So we are right now, what are we getting now, students? Right now we are getting one molecule of 3 carbon PGAL or GAP or triose phosphate. Other molecule is called as DHAP. Other molecule is called as DHAP. Now understand the concept. Your DHAP or PGAL or GAP, they are isomers. They are also again isomers. DHAP and GAP are isomers. So can I interchange them? Can I convert this DHAP into GAP? Because chemical formula is same, structure is different. So I am converting this DHAP into GAP, or I can also convert GAP into DHAP. Any one of them can happen. Any one of them can happen. Interchange can happen. That is called as isomerization. Now tell me, students, in isomerization, is there usage of energy anywhere? No. No use of energy. Energy is not evolved, energy is not consumed here. So what is happening right now? Right now, from fructose 1,6 bisphosphate, fructose, which is something like this, 6 carbon, 1, 2, 3, 4, 5, 6 carbon, 1 phosphate here, 1 phosphate here. From fructose 1,6 bisphosphate, we are getting glyceraldehyde 3-phosphate and DHAP, dihydroxyacetone phosphate. They are interchangeable. Now we are interchanging. Now, for example, for convenience, I am interchanging DHAP into GAP. DHAP is getting converted to GAP. So can I write, instead of writing DHAP separately, I am writing from this step onwards. From this step onwards, can I write glucose 1,6 bisphosphate into 2 molecules of glyceraldehyde 3-phosphate? Why 2 molecules? Why 2 molecules? Because DHAP was converted into glyceraldehyde 3-phosphate. So that is why also in your diagram you can see here, from this point onwards, everywhere it is written as 2, 2 here. 2, 2, 2, 2 and 2. Every single place. Why? Because your DHAP is now converted to glyceraldehyde 3-phosphate. Converted to glyceraldehyde 3-phosphate. From this step only, from here only. From here only. Okay? That is called as isomerization. Now students, that is the end of preparatory. You are prepared right now. Preparation is done, completed. Your particular cell is ready for payoff. Investment is done, ready for bringing the profit. Okay. Tell me, students, are you ready for payoff stage now? Ready for payoff stage? Ready for payoff stage, students? Quickly, let me in the chat. Ready for payoff? Ready? All of you. Quickly smash that like button, students, because you're understanding glycolysis, you're not by-hearting it. You're understanding glycolysis right now. Understanding it, not by-hearting any of it. Okay? Ready? Now, let's understand the next part, that is payoff right now. Payoff. So what do we have now, students? Right now we have something called as 2 molecules of glyceraldehyde 3-phosphate. 2 molecules. Why 2? Because DHAP also was converted. Because DHAP was converted to GAP. Now students, this glyceraldehyde 3-phosphate, or we can also call it as GAP, in the 5th step, GAP is getting oxidized. GAP is getting oxidized now. Tell me, what is the meaning of oxidation? Can anyone remind me? The meaning of oxidation is nothing but loss of electrons or loss of protons. All of you agree with me? Oxidation means what? Loss of protons and loss of electrons. So we are losing electrons, we are losing protons. So when this particular glyceraldehyde 3-phosphate, when it is undergoing oxidation, it is releasing electrons, releasing protons, it is also releasing the energy. Loss of electrons, loss of protons, loss of energy. Now, these two redox equivalents, electrons or protons are taken up by one more molecule. One molecule is waiting right now. All this while, chup chap, it is sitting in a corner and waiting for the chance. This electrons and protons are taken up by something called as NAD+. Nicotinamide adenine dinucleotide. Now you tell me, students, if all the protons and electrons are taken up by NAD+, addition of electrons and protons, what is it called as now? Tell me, students. Addition of protons and electrons is called as what? Tell me. Addition of protons and electrons is called as reduction. Yes. So your NAD+ will get reduced into NADH. What? It will undergo reduction. Since you undergo reduction, and when this undergoes reduction, we end up getting NADH. So that is why, students, in this step right now we have the NADH we have right now. Now what about this energy? What is the use of this energy right here? Students, remember, this energy is used to pick up. This energy is used to pick up. Disconnected, huh? This energy is used to pick up inorganic phosphate from the cytoplasm. Inorganic phosphate from the cytoplasm. Okay, it is used to pick up the inorganic phosphate from the phosphate, and this phosphate right here is now attached to the first carbon. First carbon right here. So what do we get now, students? Glyceraldehyde 3-phosphate. Can I write as glyceraldehyde 1,3? Can I write it as glyceraldehyde can be converted to now 1,3 bis phosphoglyceric acid now? See, before it was only 1. Now we are writing it as 1,3 bis phosphoglyceric acid, that is BPGA. All of you understood the concept. I will repeat again this step. Understand this. We learned just only one step, the concept of it now, like how we are getting this NADPH here, so NADH here, and how we are getting 1,3 bis phosphoglyceric acid. This step we learned. So if someone asks you a question, where does the oxidation takes place? In this step right here, oxidation. So I'll repeat again this step alone one more time. Listen very carefully. Listen very carefully, all of you. Listen very carefully. This step right here. We'll go over it one more time. So what is happening here is we have the glyceraldehyde 3-phosphate. After isomerization, we have the glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate is undergoing oxidation. When it is undergoing oxidation, it can produce electrons, protons and energy. Electrons, protons and energy. The electrons and protons are redox equivalents. They are taken up by NAD+. Oxidation, loss of electrons and protons. These protons and electrons are taken up by NAD+, and when NAD+ takes the protons and electrons, it is getting reduced to NADH. So that is the fate. That is the fate of electrons and protons. But what about its energy? Now this energy is now used to pick up an inorganic phosphate from the cytoplasm, and this phosphate is added to the first position. So that is how glyceraldehyde 3-phosphate, where the phosphate was present at the third position, glyceraldehyde 3-phosphate is converted to 1,3 bis-phosphoglyceric acid. Bis-phosphoglyceric acid right here. This step. And this step right here, since we had 2 molecules of glyceraldehyde 3-phosphate, So if one glyceraldehyde will give you 1 NADH, if one glyceraldehyde 3-phosphate is giving you 1 NADH, how many will be given by 2? Tell me. How many will be given by 2 then? Understand? So glyceraldehyde 3, if one is giving you 1 NADH, so can I tell from this point onwards, there are 2 molecules of NADH now, because there one from here and one from here. So from this point onwards, 2 NADH, not 1, 2 NADH. Clear? That is why we also have 2 here. Can you see? 2 1,3 bis-phosphoglyceric acid. 2 triose phosphate, that is basically 3 phosphoglyceric acid. Clear? So 2 of them. Now, next step is what? Right now we have the 1,3 bis-phosphoglyceric acid. Now students, tell me, I told you, where is the energy stored? Tell me students, where is the energy stored? All of you know the energy is stored in the bonds. Energy is stored here in the bond right here. Energy is stored here. Now tell me, what if I break this bond? What will happen? What will happen if I break this bond right here? This phosphate will come out. Energy will be released. And what if I add one ADP here? So can I tell, using the energy from the breaking the bond and using the phosphate, can I convert this ADP into ATP? Yes. ADP into ATP? Obviously. That is what is happening in the next step. That is 1,3 bis-phosphoglyceric acid is getting converted to energy is broken. See, bond is broken, energy is released, energy and this bond together are joined to here to add to one more phosphate, and we end up getting ATP. So instead of 1,3, can I tell right now we only have 3 phosphoglyceric acid? From 1,3 it became 3 phosphoglyceric acid. 1,3. 1,3. From 1,3 it is becoming just 3 phosphoglyceric acid. And if you look at the NCERT right here, some 1,3 phosphoglyceric acid is getting converted to 3 phosphoglyceric acid. Why 3 phosphoglyceric acid? Because 1 ATP is being produced here. Since it is into 2, can I write into 2? Into 2 ATP. Into 2 ATP now. Not 1, 2 ATP. Here also we have 2 NADH. Everything is into 2 from here. So right now what do we have? Right now we have the 3 phosphoglyceric acid. Only 3. Okay, only 3. Clear? Clear all of you? Are you understanding the concept here? Are you understanding the concept? Beautiful. We have 3 phosphoglyceric acid now. Now what is isomerization, students? What is isomerization? Isomerization is nothing but same but different. Same chemical formula, structure is different. So can I tell, if I take this phosphate from here, if I take this phosphate from the 3rd position, if I convert it to 2nd position, what will happen? Can I tell 3 phosphoglyceric acid can be converted to 2 phosphoglyceric acid now? That is what is happening in the 8th step. In the 8th step, 3 is becoming 2. 3 is becoming 2. So 2 phosphoglyceric acid. In the 8th step, 3 is becoming 2 phosphoglyceric acid. 2 phosphoglyceric acid. Two phosphoglyceric acid. What is it? 2 phosphoglycerate or 2 phosphoglyceric acid. If you see here, NCERT is the same thing. So isomerization, I told you, in isomerization, is there any energy produced? Energy evolved? No. So 3 to 2. 3 to 2. 3 to 2. So if isomerization is done, now that it is very, it has too much of water now. It has too much of water. Glycerate or glyceric acid, same thing. Glycerate. It is interchangeable. Okay. 2 phosphoglycerate. Okay, phosphoglycerate. Now this 2 phosphoglycerate will lose of water. It will lose water. And it gets converted to PEP. It will lose water and it will get converted to phosphoenol pyruvate. And I believe all of you remember PEP, right? From C4 cycle. Do you remember it? From C4 cycle. C4 cycle. In the mesophyll cell. In the mesophyll cell. PEP. Now, again students, PEP is here. Now tell me students, what will happen if I break this bond again? Bond again. This is PEP. PEP. What will happen if I break this bond? Energy will be released again. And this energy can be taken by this ADP to make ATP. Yes, ATP. That is what is happening. In the last step, the bond is broken. 2 PEP, phosphoenol pyruvate, is getting converted to pyruvate. And since it's broken down, added here, we have ATP formation in the last step, in the 10th step. In the 10th step also we have ATP formation. Clear all of you? Clear? So where is ATP produced, friends? ATP is produced in the 7th step. In the 7th step, 8th, 9th, 10th, just 1st, 2nd, 3rd, 4th, 5th, 6th, 5th conversion. This is 4th here, 3rd, 2nd, 1st. 10th step, we have the ATP formation. ATP formation in the 10th step. Do you realize? Now, before we take the break, wait. I will teach you something even more interesting. Before we take the break, I will teach something very more interesting. Wait. Understand? This is the entire 10 steps, students. Entire 10 steps. Right? Now, I will revise the entire thing one more time. Listen to me. Any doubt anywhere, you can always pause and ask. Okay? Ready for the quick revision? I'll drink some water. I'm also feeling a little dehydrated. I'll give you a quick revision. Ready for the quick revision, all of you? Students, trust me, you can just by heart this in 5 minutes and forget in 5 minutes. You can by heart it in 5 minutes and forget in 5 minutes, but understand and appreciate what glycolysis is, right? You'll never forget. So I know if you are learning glycolysis like this for the first time, it might be a little overwhelming and you'll be like, oh, so confusing, so complicated. But if you truly appreciate the beauty of glycolysis, you will never forget it. All the DNA students, if you're learning for the second time, trust me, tell me, aren't you understanding it better now? Aren't you understanding it better now already? Yes. So this is the beauty of glycolysis. The more you read, more you understand. There's no point in by-hearting. Okay? Let's do it one more time. Let's do it one more time from the start. Let's go. Show some energy in the chat because you are learning something new and not by-hearting it, students. Show some energy in the chat, all of you, quickly. Okay. Glycolysis. I'm going to revise the entire thing for you in 15 minutes. Right? I'll teach you the entire concept by concept, revise one more time in 15 minutes. Stay with me. After that, we will tell you do something called as net profit. Then I'll teach you fermentation topic. Then we will do questions. Why questions? Because only when you do questions, you appreciate the topic. If you don't solve questions, you'll be like, I understood something, I forgot something. So understand and solve questions also. Okay. Yes, ready. So what is the meaning of glycolysis, students? The meaning of glycolysis is that glyco means sugar, lysis means breaking of sugar, glycolysis. Location is cytoplasm. How many steps? 10 different steps are here. Yes. It's called as partial oxidation of glucose. Why partial oxidation? Because did you see anywhere CO2 was evolved? No. CO2 was never evolved. That's why it's called as partial oxidation of glucose. Yes. Now, in which organism does it happen? In all the organisms. Does it require oxygen? No. Your glycolysis does not require oxygen at all. It can happen in both aerobic as well as anaerobic organism. Aerobic and anaerobic, both. It can happen in prokaryotes, it can happen in eukaryotes. It can happen in plants, it can happen in animals. Clear? Now, next thing. Next thing is going to be oxidation and reduction. You need to understand, reduction is gain of electrons, oxidation is loss of electrons or protons, gain of electrons or gain of protons is reduction. It is also called as EMP pathway. It is called as EMP pathway because the scientist who discovered glycolysis is Emden, Meyerhoff and Parnas. 3 different scientists who discovered the glycolysis. The first step in glycolysis is called as preparatory stage. Then we have payoff stage. Preparatory stage, we prepare products. Payoff, we are getting the investment back. You put the money, you get the back the money. 20 दिन में पैसा double. Basically that concept. Okay. You're adding the money, you're getting back more money. Okay. Preparatory. Now in preparatory stage, the first step and third step. The first step and third step is called as what? Phosphorylation. What is happening in phosphorylation? We are taking glucose, 6 carbon right here. Glucose 6 carbon. We are taking one ATP, breaking the bond, taking the phosphate and adding it to the 6th position. So glucose is isomerized in the presence of enzyme called as hexokinase. Glucose is becoming glucose 6-phosphate. So in the first 6th position, we are adding a phosphate. So glucose to glucose 6-phosphate. Glucose to glucose 6-phosphate. I can understand Tamil, don't worry. Okay. So glucose to glucose 6-phosphate. Clear? First step done. Using of ATP, yes, ATP is used. ATP is used in the first step. Okay. Now, isomerization. Same but different. Same but different. What is happening here? Isomerization means the chemical structure is different. Chemical formula is same. Your glucose is phosphate and fructose is phosphate. Both of them have same chemical formula but different structure. See here. Your glucose and fructose, they have the same chemical formula, C6H11O9P, but structure is different. Here we have an aldehyde, here we have a ketone. So can I interchange? Yes, I can interchange. So glucose 6-phosphate is getting isomerized into fructose 6-phosphate. Glucose to fructose 6-phosphate. Since it is isomerization, there is no requirement of oxygen, there is no requirement of energy. Second step is done. Here. Second step done. Now what is happening in the third step? In the third step, again phosphorylation is happening. Phosphorylation is happening. Okay, understand. To this glucose 6-phosphate, If I take one more ATP and break the bond, use the energy, use the phosphate and adding it to the first position, what will happen? Your fructose 6-phosphate, can I call it now fructose 1,6? Yes. So what am I getting now? We are getting from fructose 6 to fructose 1,6 bisphosphate. Again, our ATP is used. Where? In the first step and in the third step. Two different steps, ATP is being used. Now, fructose 6-phosphate we have, fructose 1,6 bisphosphate we have, which is 6 carbon. 6 carbon. I am breaking the 6 carbon molecule into a 2 molecules of 3 carbon. 6 carbon, breaking it into 2 molecules of 3 carbon. So we have one more. What are those? These two. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. DHAP and GAP or PGAL. Same meaning. Okay. See here. We have glyceraldehyde 3-phosphate, PGA or GAP or triose phosphate. It can also be, the other one is called as the DHAP, dihydroxyacetone phosphate. Now both of them are also isomers. We can interchange among them. So what are we doing? We can interchange. So we are converting convenience. DHAP is now converted into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. DHAP is now converted to glyceraldehyde 3-phosphate, all of you. Now, this glyceraldehyde 3-phosphate we have. Glyceraldehyde 3-phosphate. How many? 2 of them. Why 2 of them now? Because DHAP is now also converted back. So 2 molecules of gap. Now students, this gap right now undergoes oxidation. Payoff stage starts now. Till here it is preparatory. Fourth step. Fifth step. No, this was fourth here, right? First, second, third, fourth, fifth, and sixth step here, right? 6, 7, 8, 9, 10. Yes, sixth step. Right? Sixth step. So now sixth step is happening. So what is happening? Your glyceraldehyde 3-phosphate undergoing oxidation. Now what is oxidation, students? Oxidation is nothing but loss of electrons or loss of protons. So this is undergoing oxidation right here. Oxidation right here to produce the redox equivalents. Electrons, protons, energy. Protons and electrons are taken up by your thief. Who is the thief here? NAD+. NAD+ is taking the protons and electrons and it is getting reduced. Can you see? It is getting reduced to NAD+. And that is how, students, that is how we get the NAD+ there in the sixth step. Sixth step, NAD+. Beautiful. Now what about the energy? This energy is now used to pick up an inorganic phosphate from the surrounding and this phosphate is now attached here. So can I tell glyceraldehyde 3-phosphate, can I call it 1,3 now? See, 3-phosphate 2, can I call it now 1,3 bisphosphoglyceric acid? 1,3 bisphosphoglyceric acid. This step right here. And 1,3 bisphosphoglyceric. And can we also have NAD is produced? Yes, NADH is produced as a result of reduction. 2 molecules, because everything is 2 from here. Now we have 1,3 bisphosphoglyceric acid. Now I'm breaking the bond. Bond is broken, energy is released, energy is taken up by NADP, NADP is converted to ATP. This step right here we have a ATP. How many? Into 2. Okay. 6, 7, 8, 9, 10. 6th step, 7th step. So remember, students, ATP is formed in the 7th step and 10th step. 2 different steps, ATP is produced. I'll give you a break after this. Okay. 7th step and 10th step. Okay. ATP is being produced. Okay. Now 3-phosphoglyceric acid is again undergoing isomerization. Your 3 is becoming what? This is all done, no? 3-phosphoglyceric acid is undergoing isomerization. 3 is sent here, 2. 3 to 2. So we're getting 2 here. So 3-phosphoglyceric acid is getting converted to 2-phosphoglycerate. Isomerization. Now to this isomerization, I am removing water. So once you remove from an acid, if you remove the water, it is getting converted to phosphoenol pyruvate, PEP. Phosphoenol pyruvate. Phosphoenol pyruvate. All of you understanding, right? What is being removed? Now we have ninth step. This is a ninth step. We get phosphoenol pyruvate ninth step. Now, from this phosphoenol pyruvate, we have one more phosphate left here. One more phosphate left here, PEP. Now, what if I break the bond again and energy is released? Can I use the energy to add to an ADP and to make the ATP? Yes, ATP. So, in the last step, we get pyruvic acid or pyruvate with the release of again 2 ATP. So, seventh step and 10th step, we have 2 ATPs, 2 ATP. And in the sixth step, we have the NADH production, NADH production. Where is ATP consumed? ATP is consumed in the third step and the first step. ATP is being consumed. So, clear? So, that is all the glycolysis is all about. Now, all of you can go take a break. Okay. And once you come back from the break, I will teach you what is net profit. What is net profit? And after teaching you net profit, I will also teach you something called as all the questions. And then we will learn the next concept, fermentation, also today only. And remember, students, fermentation is a very important concept. You can buy heart all this, students. You can just buy heart fermentation, but you will not ever understand. See this. This may seem very easy. This may seem very easy, but this is a very complex process where we have reduction, oxidation. It is a redox reaction. Here we have oxidation, here we have reduction. The same goes for your alcoholic fermentation also. There is reduction and there is oxidation also. So it's a very beautiful thing we are coming up later on. So don't miss that out. You can just buy heart it. It's very easy to buy heart this. Pyruvate to lactate, lactate dehydrogenase, it's done. If you just buy heart it. But in my class, there is nothing called as by heart. I want you to understand this. Okay. So for you to understand, you have to come back later on. Then we'll read the NCERT also. This is the beauty of plant physiology, students. When you understand it, it becomes very easy. Okay. So let's take a break now. What's the time? My watch is a little slow. What is the actual time? 8:13. 33. 8:35 will come back. Resume at 8:35. 8:35. Okay. So we'll resume at 8:35. All of you need some time to eat, right? All of you need some time to eat. Take easily 20 minutes you can take. Eat aram se and chill. And you can come back here and then we'll do the rest of the things today. Okay. And we have questions also. Okay then. I'll see you after exactly at 8:35, all of you. Bye-bye. Take care. So let's start the last part of today's class. That is going to be the net profit. Net profit. Okay. Net profit in the entire process, right? Net profit. Hello, 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 hello. Welcome back. Welcome back. I hope all of you ate some food, right? All of you ate some food. Yes. All of you ate food. Very good. Very good. Eat food nicely. Okay. Gaming logo editor. Oh, we edited logos, no? That is nice. Tell me, which game have you played? Can you also edit some photo? Edit some logos for me also. Okay. Free, but free of cost, obviously. Okay. Now, students, let's understand the net profit. Let's understand the net profit here. Okay. Net profit. Net profit here. Understand. Now we will divide it again. We will divide it again into depending upon the preparatory and pre-op stage. Okay, preparatory and pre-op stage. Now tell me, in preparatory stage, how many ATP is consumed? All of you know, in the first stage, step and third step, can I tell 2 ATP is consumed? 1 ATP here, 1 ATP here. So can I tell minus 2 ATP? Can I tell minus 2 ATP? Minus 2 ATP is consumed. Okay. What about in payoff stage? In payoff stage. In your payoff stage, if you look at the sixth step, in the sixth step, can you tell there is NADH? But is it 1 NADH or 2 NADH? Tell me all of you in the chat. Now here, since we have from in this point onwards, we have everything is 2. Why? Because DHAP is getting converted to gap. So can I tell in the sixth step, we are getting 2 molecules of NADH. We are getting 2 molecules of NADH. 2 molecules of NADH. And all of us know 1 NADH gives 3 ATP. So what is the answer? 3 into 2. 3 into 2. That is totally 6 ATPs here. 6 ATPs. And students, remember, this 3 and 2 is there, right? 3 and 2 is very old way to learn it. The actual calculation should be NADH as 2.5 ATP and FADH as 1.5 ATP. But NCERT is a little slow here. NCERT is a little slow here. Okay. But right now, understand, NCERT is giving 6 ATP. Now, tell me how many ATP are produced directly? Directly, how many ATPs are produced? If you look here, in the 7th step and the 10th step, 2 ATP here, 2 ATP here. Why 2? Because of multiplying here. So totally 4 ATP are directly produced. In the payoff stage, can I tell 4 ATP are directly produced? 4 ATP are directly produced. So minus 2, 6 and 4. Now tell me, students, what is 6 plus 4? Quickly in the chat. 6 plus 4 is 10 ATP. 10 ATP. 10 ATP minus 2 ATP. 10 ATP minus 2 ATP. That is around 8 ATP. That is around 8 ATP, which is called as, this right here is the net. This right here is the gross. 10 is the gross. 10 is the gross. Ten is the gross. 2 is the net. All of you understood? Everyone in the chat? In the examination, they can directly ask you, what is the net gain of ATP? What is the gross gain of ATP? Now, students, ready for some questions. Which type of question can be asked here? Which type of question can be asked from this glycolysis concept? I will show you the questions later on, but let's try to tackle some questions now. Now here, I'll teach you something called as the irreversible steps. Irreversible steps. They can ask you about irreversible step. So in the entire cycle here, the first step, third step and the 10th step, 3 different steps are irreversible. First, third and 10th. These 3 steps are irreversible step. Irreversible step. They can ask you a question like this. Or they can ask you, students, which is rate determining step, means the slowest step. Rate determining step. Rate determining step. What is the answer? The rate determining step here is the third step. Third step. Phosphorylation. This right here is the rate determining step. Third step. Third step is the rate determining step. They can ask you one more question. Which step water is removed? Which step water is removed? You will see the last but one step. That is the ninth step. Can you see? In the ninth step, water is being, in the eighth step, water is being removed. Eighth step, ninth step only. Yeah, ninth step only. This ninth step is coming down is water is being removed. Ninth step, water is being removed. So you can write down, students, water is being removed in the ninth step. Or can I write directly? Can I write directly from PG? Can I write from 2PG into PEP? 2PG into PEP. 2PG phosphoglycerate into phosphoenol pyruvate. Phosphoenol pyruvate. Okay. Now they can also ask you a question something like in which step NADH is produced? NADH is produced. NADH is produced. See, sixth step, you see, sixth step, PGAL. Sixth step, if you see here, sixth step, if you see here, sixth step, if you see, PG or gap is getting converted to here bisphosphoglyceric acid. So can I write in the sixth step? In the sixth step. Okay. That is phosphoglyceric acid is converted to 1,3 bisphosphoglyceric acid. 1,3 bisphosphoglyceric acid. Okay. I'll just put a box around it. Okay. Okay. That is what is happening here. Okay. Or they can ask you which step ATP is consumed. Which step ATP is consumed? You will tell first and third. That is glucose to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. First and third step. Can you see? In the first step, glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. ATP is consumed. And then they can ask you one more question. Which step ATP is produced? You will tell, sir, 7th and 10th. 7th and 10th step. Can you see here? In the 7th step, we have the 1,3 bisphosphoglyceric acid to 3-phosphoglyceric acid. 1 ATP was produced here. And also in the last step, we have phosphoenol pyruvate to pyruvate. 7th and 10th step. Beautiful. 7th and 10th step. Clear? Irreversible reaction done. Which step ATP is consumed done? What is removed? NAD is produced. Yes. Now they can all sometimes ask first enzyme in the reaction. That is hexokinase. The first enzyme, students, the first glucose to glucose 6-phosphate, the enzyme is the hexokinase. Okay. Or they can ask you last enzyme. Only these two will ask you. Last enzyme is pyruvate kinase. Pyruvate kinase. Last enzyme. Last enzyme is between PE to pyruvate. PE to pyruvate is pyruvate kinase. Last enzyme is PE to pyruvate kinase. Then all of these questions can be asked from the glycolysis. All of these questions can be asked from the glycolysis. Okay. Or I can ask you, students, why it is called as EMP pathway. Why it is called as EMP pathway. Or I can also ask you oxidation which step. Oxidation which step. Which step. Or they can also ask you which step of the entire reaction, right? What was coming in my mind, I forgot now. Does it happen in aerobic or does it happen in anaerobic? Aerobic or anaerobic? What is the answer? Tell me, students. It happens in aerobic or anaerobic? Answer should be both. Both. Happens in both. There is no discrimination. Happens in both. So students, do you realize all possible questions? Like I have showed you some questions here. I have shown you some questions here. Obviously, you can make more questions depending upon the location, partial oxidation, right? How many steps are there? So all of these questions can be asked from the entire glycolysis. Are you satisfied with glycolysis, students? Tell me in the chat. Are you satisfied with the glycolysis the way I taught you? Now, yes, this may be the first time you're trying to learn glycolysis in such a fashion, but understand, students, when you learn the glycolysis, the concept wise, right, you will never regret it. The way you have spent your time with me right now and understanding it conceptually and not by hearting it, you will truly appreciate it when you go to MBBS first year, because that's when you can't by heart anymore. You can't by heart anymore. You have to understand it. Okay, beautiful. Understanding all of your questions can be asked. Let's read what is given in NCERT, all of you. Let's read what is given in NCERT. Okay, let's read what is given in NCERT. Okay. The term glycolysis as originated from the Greek word glykos. Glyos means sugar, I told you in the starting, and lysis means splitting. So sugar is being split open here. Sugar is being split open. Yes. The scheme of glycolysis was given by the Gustav Emden, Otto Meinhof, and J. Parnas, and often referred to as EMP pathway. That is what it is called as what? EMP pathway. Okay, EMP pathway. In anaerobic organisms, it is only processed in respiration. Students, remember, your entire glycolysis can take place in anaerobic also. And in anaerobic respiration, it's one of the only respiration pathways. Okay. Glycolysis occurs in cytoplasm. Location is cytoplasm. Yes. Location is cytoplasm right here. Location is cytoplasm of the cell and is present in all living organisms. So all living organisms can undergo glycolysis. There is no discrimination. Aerobic, anaerobic, prokaryotic, eukaryotic, plant, animal cell, everywhere. In this process, glucose undergoes partial oxidation. Partial oxidation. What does the meaning of partial oxidation, students? CO2 is not evolved. Partial oxidation will give you pyruvic acid. That is the end product. End product. In plants, this glucose is derived from the sucrose. I told you. With the help of which enzyme, students? With the help of enzyme called as invertase. With the help of enzyme called as invertase, it is getting converted. Right. End product photosynthesis. Two are from the storage. Okay. Carbohydrates, sucrose is converted to glucose and fructose by the enzyme invertase. Okay. And these two monosaccharides are readily enter the glycolytic pathway. So two of them can also enter. So fructose can also enter now, because simple sugar now monosaccharide. Glucose and fructose are phosphorated, the first step, to give rise to glucose 6-phosphate. The first step, glucose to glucose 6-phosphate, phosphorylation is happening. ATP is used. Yes, ATP is used. By the activity of enzyme hexokinase. See, so learn hexokinase. Hexokinase is important. This phosphorylated form of glucose is then isomerization, second step, to produce fructose 6-phosphate, isomerization to fructose 6-phosphate. Subsequently, the steps of metabolism of glucose and fructose are same. The various steps in glycolysis are depicted in the figure. In glycolysis, a chain of 10 reactions. See how many steps? 10 steps. 10 steps. Under the control of different enzymes takes place in the to produce pyruvate from glucose. While studying the different steps of glycolysis, please note that the steps which utilize or synthesize ATP and NADH takes place. So understand where is ATP is being produced, where NADH is being produced. We already know that now. We already know that. Right. ATP is utilized in the first two steps. I told you already. It is actually first and third, because second step nothing is happening. Okay. First in two steps, not first two, in two steps. First in the conversion of glucose to glucose phosphate, and second in the conversion of fructose to 6-phosphate to fructose 1,6 bisphosphate. In these two steps, that is the first step and third step. First step and third step, it is being consumed. First step and third step, it is getting consumed. Okay. Now, ATP is utilized in the first two steps. All of you learned that. Now, fructose 1,6 bisphosphate is split into two molecules. That is DHAP and PGAL. Okay. We find that there is one step where NADA plus is formed from NAD plus. Reduction. This is when the 3-phosphoglyceric aldehyde PGAL is converted to BPGA, this one. 1,3 bisphosphoglyceric acid. 1,3 bisphosphoglyceric acid. NADH is being produced. That is the sixth step. Two redox equivalents are removed in the form of hydrogen atoms. We learned this, remember, from PGAL, and transferred a molecule to NADH, NAD plus. All of you remember the reduction process. Reduction, it's given here. Reduction is happening. Yes, reduction is happening. The PGAL is oxidized and with inorganic phosphate to get, see, remember, the energy is taken. I told you, the energy is used to bring one inorganic phosphate and converted. We learned that here also. To get converted to BPGA. The conversion of BPGA to 3-phosphoglyceric acid, PGA, is also energy yielding process. It's an energy yielding process. It means that this step, BPGAL, 1,3 bisphosphate is getting converted to 3-phosphoglyceric acid. ATP is being produced. Energy yielding process right here. Energy yielding process. This energy is trapped by the formation of ATP. I told you how ATP is formed. Okay. Now, another ATP synthesized during the conversion of PEP. Last step. Phosphoenol pyruvate to pyruvic acid. The last step also, one more ATP is produced. So ATP is produced in the 7th step right here, 7th step and 10th step. 7th step and 10th step, ATP is produced. One ATP here, 7th step, 8 and 9 nothing, 10th step again ATP is produced. So ATP is produced in two steps, 7th and 10th, consumed in 1 and 3. 6th step NADH, 6th step NADH. Here. Can you then calculate how many ATP is produced? We calculated, done. Pyruvic acid is then key product of glycolysis. What is metabolic fate of pyruvate? We'll understand now. What is the fate of pyruvate? What is going to happen in pyruvate? Okay. Three major ways in which different cells handle pyruvic acid. Three different ways. Let's understand the fate of pyruvate now. What is the fate of pyruvate? Understand, students. This you have learned in the class 10th also, I think. In class 10th, if you're from CBSE syllabus, in the chapter Respiration in Plants or Animals, I don't know what is the chapter name, you will have a table called as Respiration. You have a table which says fate of pyruvate, right? Fate of pyruvate. For example, we have glucose here. Glucose is getting converted to pyruvate. Glucose is converted to pyruvate. All of you know that glucose is converted to pyruvate. That is 6-carbon glucose. 6-carbon glucose is converted to 3-carbon pyruvate. This conversion is happening. Right here. And what is this conversion called, students? This conversion is called as glycolysis. We call this glycolysis. And where is it happening? This is happening in the cytoplasm. So glucose is converted to pyruvate in glycolysis. This is called as your pyruvate. We're getting pyruvate in the end. Now this pyruvate can undergo three different things now. This pyruvate right here can undergo three different ways. First one, second one, third. Three different ways. The first one is, first two are in absence of O2. Absence of oxygen. Absence of oxygen. The last one is happening in the presence of oxygen. The first one is happening in the case of your yeast cell. This one is happening in the case of your muscle cell. The last one is happening in the case of mitochondria. In the mitochondria. Now, when it is happening in the yeast cell, we are getting something called as ethanol plus CO2 plus energy. We are getting ethanol plus CO2 plus energy. Now, what about in the case of muscle cells? In the case of muscle cells, you might have seen you get a cramp sometimes. You get a cramp suddenly. In the case of muscle cell, we get lactic acid. Lactic acid plus energy. In the case of your mitochondria, we get CO2 plus H2O plus energy. Okay, CO2 plus H2O plus energy. The first one, which is happening in the yeast, we can call it alcoholic fermentation. The second one is called as lactic acid fermentation. Third one is normal oxidation. Normal simple oxidation or aerobic oxidation, or also can also call it aerobic respiration. So the three different fates of your pyruvate. Glucose is converted in cytoplasm in glycolysis pyruvate. Pyruvate, in the absence of oxygen in yeast cell, it can undergo ethanol, CO2 and energy, alcoholic fermentation. In the case of muscle cell, it is undergoing lactic acid and energy. It is called as lactic acid fermentation. Third one is called as in presence of oxygen in mitochondria, it is getting converted to CO2, H2O plus energy, called as aerobic oxidation or aerobic respiration. This is called as the fate. This is called as fate of pyruvate. See here, students. Pyruvic acid by glycolysis. The first one is glycolysis, right? We're getting glycolysis. These are lactic acid fermentation, we learned just now. Then the second one, you have the alcoholic fermentation. And third one is called as aerobic respiration. Three different pathways. Fermentation takes place under the anaerobic conditions. I told you, absence of O2 in many prokaryotes and unicellular eukaryotes. For a complete oxidation of glucose, CO2, H2O. However, organism adopts Krebs cycle, which is also called as aerobic respiration. It requires O2 cycle. So can I tell students, the last step we told here, this is happening in the Krebs cycle. The last one. It is happening in the Krebs cycle. So this is a beautiful thing which is happening, called as the fate of pyruvate. What is happening to the pyruvate in general? Okay, fate of pyruvate, all of you. Now students, now we go to the next concept. What have we done right now? We learned about respiration. We are now focusing on anaerobic respiration. Now we will focus something called as lactic acid and alcoholic fermentation. Ready? I know students, the session is going a little longer. Why? The reason I have told you always, right? The reason I'm doing long session is to make sure that every detailed concept is learnt now only. Can we go this detailed when the exam is near? No, we cannot do that. We cannot do that when the exam is almost near. So we are going as detailed as possible now only, so you don't have to suffer later on, right? Now, for example, let's understand lactic acid fermentation. Lactic acid fermentation. First of all, tell me, where does it take place? Tell me, students, where does it happens? Lactic acid fermentation takes place in the case of your muscle cells. Muscle cells, lactic acid formation, muscle cell. Now understand here what is happening. Pyruvate is here. Yes, pyruvate is right here. Okay, we have pyruvate here. Okay, pyruvate is here. Okay, this pyruvate is the first electron acceptor. Now it has to get electrons from somewhere. It has to get electrons from somewhere. Understand this concept. Small concept is here. Small concept is here. Now, students, in the first step, your NAD plus NADH, it is undergoing oxidation. It is undergoing oxidation. And tell me, what is oxidation meaning, sir, students? What is the meaning of oxidation? Meaning of oxidation is what? Loss of electrons or loss of proton. Loss of. Now, there's electrons and protons. Electrons and protons which are being lost, they are taken up by pyruvate. They are taken up by pyruvate. So, can I call them first electron acceptor now? Yes. First electron acceptor. Why? Because they are accepting the electrons from oxidation of NADH. Oxidation of NADH. So, NADH is getting oxidized. So, can I tell we get NAD plus because it lost the protons and lost the electrons? NAD plus. Yes, NAD plus. Okay. So, we get pyruvate. Pyruvate is accepting electrons. Now, students, tell me, if pyruvate is accepting the electrons and accepting the protons, can I tell now pyruvate is undergoing reduction or oxidation? Tell me. If pyruvate is accepting the electrons and accepting the protons, can I tell pyruvate is reduced to lactate? Lactate. It is reducing to lactate. And the enzyme involved here, which can be asked as a famous question all the time, the enzyme here is lactate dehydrogenase. Lactate dehydrogenase. Lactate dehydrogenase. That is the first concept which you need to understand. NADH is undergoing oxidation, loss of protons. The protons are taken up by pyruvate, first electron acceptor. It is getting converted to lactate. Enzyme involved is lactate dehydrogenase. That is the entire concept of lactic acid fermentation. Similarly, you need to understand what is called as alcoholic fermentation also. What is alcoholic fermentation? Understand, simple again. In alcoholic fermentation, your acetaldehyde right here is the first electron acceptor. Why, sir? Understand. Now here we have pyruvate. The first enzyme involved here is called as your pyruvate decarboxylase. See, students, in the previous step, 3 carbon, there was no change in the carbon number. Carbon number was maintained as the same, 3 carbon and 3 carbon. But in the second step, in the alcoholic fermentation, there is decarboxylation. So, one CO2 is evolved. So, do we not learn, students? Do we not learn in the alcoholic fermentation? Do we not learn in the alcoholic fermentation? In the yeast cell, CO2 is evolved here. Can you see? CO2 is evolved. That has to happen now. That is happening here. Can you see? So, CO2 is being evolved because of pyruvate decarboxylase. CO2 is being removed. Understand the concept. You can by heart all this later on. CO2 is being revolved. So, once CO2 is being revolved, we get something called as acetaldehyde, which is just 2 carbon. Acetaldehyde, 2 carbon. Now, what is happening again? Again, what is happening here is your NADH is getting oxidized. NADH is getting oxidized here. If NADH is getting oxidized, can I tell it is losing electrons or losing protons. Yes, electrons and protons are being lost here. So can I tell this electrons and protons are taken up by acetaldehyde? Yes, they are taken up by acetaldehyde. If they are taken up by acetaldehyde, can I tell acetaldehyde is the primary electron acceptor now again? Yes, acetaldehyde is the primary electron acceptor. And the enzyme involved here is called as alcohol dehydrogenase. Like how we had pyruvate lactate dehydrogenase here, similarly we have the alcohol dehydrogenase. And since your acetaldehyde is receiving the protons from here, receiving the electrons from here, can I tell acetaldehyde is reduced? Acetaldehyde is reduced into ethanol. It is reduced into ethanol now. Concept. It is reduced into ethanol. Primary electron acceptor. Reduced into ethanol. Okay. That is the concept. Let's read it. In fermentation, say yeast, the incomplete oxidation of glucose. See, incomplete oxidation. Why it's called incomplete oxidation in fermentation? Because only some amount of CO2 is released. That is incomplete oxidation. Some amount of CO2 is being released. Okay. Of glucose is achieved under anaerobic conditions by a set of reactions where pyruvic acid is converted to CO2 and ethanol. CO2 and ethanol. Since some amount of CO2 is still used, some amount of CO2 is getting converted, some amount of glucose is converted to ethanol, some amount of pyruvate is converted to ethanol, some amount is coming as CO2. So partial oxidation. So that's what's called as incomplete. What is partial? Partial was no CO2 evolved in glycolysis. Okay. So here, since only one CO2 is coming and some amount of pyruvate is converted to ethanol, it is incomplete oxidation. The enzyme involved here is two of them. First one is the pyruvic acid decarboxylase. The first one. The second one is called as alcoholic dehydrogenase. Two enzymes in alcoholic fermentation. In the lactic acid fermentation, in lactic acid fermentation, catalyzed by reaction. Other organisms like bacteria produce lactic acid, muscle cells, or from pyruvic acid. The steps involved is very simple here. In animal cells also, in muscle cells, when the oxygen is inadequate, when there is no oxygen at all, during cellular respiration, pyruvic acid is reduced. See, I told you, pyruvic acid is reduced. Didn't we learn that? Here? Pyruvic acid is reduced into lactate, reduced into lactic acid by lactate dehydrogenase enzyme. The reducing agent, I told you, the reducing agent who's taking up who's giving the electrons and protons is the NADH. It is the NADH here, which is reoxidized to NAD plus, So NADH is getting reoxidized to NAD plus. Now, what is the meaning of reoxidize? The meaning of reoxidize is because remembering glycolysis, because of oxidation, we got NADA. And because of oxidation, we got NADH in the sixth step. In the sixth step, because of oxidation, we got NADH, right? In glycolysis. That is why it is mentioning reoxidize now. Reoxidize to NAD plus in both process. See here. In your glycolysis, students, in the glycolysis, we got NADH because of oxidation. We got this. Now, what is happening here? In pyruvic acid is getting converted to lactic acid. In lactic acid fermentation, it is being used again. This NADH is used here. It is completely used here. Reoxidized to NAD plus. Same goes for your alcoholic fermentation. In alcoholic fermentation also, whatever NADH is produced here in glycolysis, this NADH is again used in the case of alcoholic. So NADH is again converted to, it is reoxidized to NAD plus. So can I tell this NAD plus will again go back here? Yes, it can again go back there. That is the beauty of fermentation. NADH is being reused. Okay, and we got ethanol and CO2 here, we got lactic acid. Now students, net gain here. What is the net gain? Here? Net gain of ATP. So in preparatory stage, again, same glycolysis, right? Same glycolysis happening. In preparatory stage, we have minus 2 ATP, minus 2 ATP. In payoff stage, in payoff, 2 NADH is produced. But can I tell this 2 NADH is being consumed here, right? Reoxidized, it is consumed here. So I'm gonna not mention that it's consumed now. Now again in preparatory stage only, in payoff stage, we are getting 4 ATP at the 7th step and the 10th step. 7th step and 10th step, we are getting ATPs. So you have 4 ATP. So what does the net gain tell me? Net gain. Net gain, since your NADH is not there, we only have ATP. 4 minus 2, tell me. 4 minus 2 is 2 ATP. 2 ATP. But in the case of normal glycolysis, we have more ATP. 38 ATP are produced in normal aerobic respiration. So didn't we learn in the starting, when I was looking at the difference between aerobic and anaerobic, we learned there, right? In aerobic respiration, more energy is produced. But in anaerobic respiration, very less energy is produced. Just 2 ATP is produced. Just 2 ATP is produced in anaerobic respiration. Just 2 ATP is produced in anaerobic respiration. All of you, I believe, understood that point. And students, if you're able to understand this concept right, and you're not by-hearting it, quickly smash that like button, all of you. Quickly smash that like button and let me know in the comment section if you truly enjoyed respiration by not by-hearting it. Right? See here. In both lactic acid and alcoholic fermentation, not much energy is released. Not much energy is released. Less than even 7% of the energy in glucose. So very less energy. How much less? Less than 7% of energy in glucose is released, and not all of it is trapped as high energy bond in ATP. See, less than 7%. Less than 7% is used. That's all. Also, the process are hazardous. Even either acid or alcoholic is produced. Very dangerous, right? Very dangerous. Very dangerous. Either acid or alcohol is produced. What is the net ATP? We told you 2 ATP. Okay. Now ATP utilized during glycolysis. 1 molecule of glucose is fermented to alcohol lactic acid. 2 ATP we told you. Now yeast poison themselves to death. Yeast will kill itself. No, suicide. When the concentration of alcohol reaches about 13%. That is why they tell. That is why they tell in the normal, if they make beer, normal beer, it cannot exceed more than 13%. If an alcohol is prepared naturally, the percentage of alcohol it can have maximum is 13%. It cannot go more than that. Cannot go more than that. If it goes more than 30%, because yeast will die. Then you might ask me, sir, what about the alcohol in which they say 40%, 70% alcohol, 30% alcohol? That is because of distillation. That is because of distillation. They remove more water, they evaporate water, and alcohol content increases. Distillation. Beer, the beer drink alcohol. So alcohol, that is beer, can have only 13% maximum, not more than that. Beer can have. Okay. That's what. How would you be maximum concentration of alcohol in beverages other than the naturally fermented? That can increase more than 20, 30, 40, 50, 60, 70. That is because of distillation. How do you think alcoholic beverages? I told you already this. What is then the process by which organism carries complete oxidation? How do organisms carry complete oxidation now? Complete oxidation is happening because of aerobic respiration. Because of aerobic respiration. Aerobic respiration. In the process that leads to the complete oxidation of organic substance. Students, remember, glycolysis. Glycolysis, see, beer alcohol is between 3 to 6, but maximum beer can be 13. That's all, not more than that. Usually between 3 to 8, 9, 10 also is there. They make this thing, crafted beer up to 10%, but 13% is the maximum. Okay. Students, remember, glycolysis is partial oxidation. Fermentation is incomplete oxidation. Glycolysis is partial oxidation. Fermentation is incomplete oxidation. But aerobic respiration is complete oxidation. Aerobic respiration is complete oxidation of organic substance. Aerobic respiration. in the presence of oxygen, and release of CO2 and water, and large amount of energy is present in the substrate. This type of respiration is most common in higher organisms, plants and animals. Plants and animals. I think we will stop there today. Okay. Aerobic respiration we learned. This is your aerobic respiration. Aerobic respiration. So all of you know pyruvate is produced. All of you know pyruvate is produced in the cytoplasm. In the cytoplasm. Now students, pyruvate is taken and it is transported within the mitochondria. It is sent inside the mitochondria. See, we are sending inside the mitochondria. Now once pyruvate goes inside the mitochondria, there are multiple steps it has to go through. It has to go multiple steps. The multiple steps are called as what? Acetyl-CoA, Krebs cycle, electron transport chain. All three we will learn in next class. Acetyl-CoA, what is happening? That is oxidative decarboxylation. We will learn in next class. In the next class, we will learn something called as Krebs cycle. Then we will also learn something called as oxidative phosphorylation. Three different steps happening in the aerobic respiration. Three different steps happening in the aerobic respiration. This is the syllabus. Yes, obviously I can understand Telugu and English and Hindi and Telugu and Kannada, right? So next step will be we'll be learning about aerobic respiration, mitochondrial oxidative carboxylation, Krebs cycle, and electron transport chain in the next class. In the next class, okay. Now we finish. So thank you so much, students. I know today was a long class. I have taken as detailed as possible. This chapters can go, this topic can go through, right? You have done the detailed version of it. So let me know in the comment section if you enjoyed today's class, and if you want such classes, or we can do easy. We can always do, I can take glycolysis, teach you a trick and finish it in 10 minutes. I can finish the entire glycolysis in 15 minutes and finish it off. I don't mind doing that also. Why do you think we stretched it out? Why do you think we stretched it and make sure you understand it? Because it is beneficial for our students to not by heart it and not by heart it, right? That was the main goal for today's class. And hopefully all of you understood in today's class. Students, before I go, let me tell you, whatever you're doing right now, right? If you want to get into any of the Vedantu's paid batches or offline batches, and you do not have the money to do it because of some financial conditions, you're not able to do it, and you really want to get into Vedantu's paid platforms also because they're really good. How do you do it? There's an exam called as MVSAT exam just coming up right now, and if you are really good at what you do, you're smart and genius, write the exam, and let's see what happens. If you get good marks or good rank in that, you can get up to 90% off on the paid batches. And I'm not making it up, it is a reality. So write the MVSAT exam, register for the MVSAT exam right now. Link is in the description. So this is my humble request for all of you. Physics, chemistry, maths, bio, everything is there. Not only one, everything is there in that. Okay, so do that. You can always get iPhone, everything is there, but that is the main important part. Main part is you getting into Vedantu's platforms for less money. Okay, so my work done here. My work is done here, students. And now it's up to you to request all the students to smash the like button. Let me know how the session was in the comment section. Let me know in the comment section, all of you. I will check the comment section. I couldn't give you a homework question today because I want to see your response in the comments today. And I want to see how much you love today's session and how much you love the botany from your sir. Right. So I hope the botany is more interesting now. It is not simple by hearting anything. So thank you so much, students, for attending for 3 hours, 20 minutes class, and I will see you tomorrow at 6 p.m. Yeah, tomorrow 6 p.m. we'll finish the class. We'll finish the chapter off completely. We'll kill the chapter tomorrow. Tomorrow only. Let's not have a large break. Let's not have a big break, right? So we'll break it off. We'll finish the chapter tomorrow only. Thank you so much, students. Bye bye. Take care, all of you.